All right. Speaking of which, let's talk about the exams first. We had the grades were posted last night after a Herculean effort on my part. I was able to get everything graded and curved and input. As you can see, the averages aren't great for these first exams. They're a little lower than we would certainly like them to be for a first exam. But as you can also see, again, I've posted the two high scores and the two low scores, not to make two people feel really good about themselves and two people feel really bad about themselves, but to remind you that there's more to numbers than just an average. This first exam typically has an incredibly wide range of scores to them, because people come into this class with different expectations, especially now that we're online. There's some people who have completed some, or even in some cases, most of 430 already. Have taken other science classes like micro and things like that and understand what is expected of them in a class like this. And so they come in a little bit better prepared. Whereas other people, despite all my warnings, despite all the doom and gloom I speak at the beginning of this, don't fully appreciate what is expected of them and how this class is different from most of the classes you've ever taken before. So we have these wide scores. Again, we talked about this a little in the last class. Obviously, the people who did really well, does this mean they can just rest on their laurels from the rest of the class and just glide into an A at the end of the semester? No, of course not. Those people are still going to have to work really hard to continue to do what they're doing. The good news is they can feel confident in the fact that they're on the right path, and hopefully if they can continue to do what they've been doing, they will continue to be successful. By the same token, the people who were not as successful as they would want to be on this test shouldn't, grab their whip and go flog themselves as a result of this or feeling bad about it. Use this first test as a learning experience to help you to understand what is going to be expected of you in this class and how best to tackle this material. Because the good news is the same way I wrote these first lab and lecture exams is how I'm going to write all the lab and lecture exams. So now you know how I'm going to ask questions, how I'm going to grade those questions, what's expected of you. And so you can use that information to better prepare yourselves moving forward. Now, there is still, while those grades are not where we would want them to be, there are two really important pieces of information. The first is that, remember, this class is based on all of your points, not just the exams. And so right now, if we look at all of the points in the class, our average is 230 out of 314, which is almost 74%, which is not a bad place to be after the first exam. And the other thing to remember is we are probably going to finish this class with somewhere between 1,200 and 1,300 points. And we're only 314 points in now. That means that there is a ton of points left. As I mentioned, mathematically, every single person in this class is still capable of getting an A. There are enough points left over. Remember, you can use your final to replace your lowest lecture score. You have all these opportunities to improve and maximize your points, make sure you're turning in your homeworks and getting them turned in on time and all the things that go along with that to get the grade that you want. Every single person can still get the grade that they want in this class. Some people are just going to have to change the way they're attacking this material better than others. Now, as I mentioned, the grades were released yesterday, but not the exams. I will be releasing the exams today. I will be releasing them after lecture. If I do it before lecture, then all of you are looking at your exams and not listening to my lectures. So it is important that we pay attention to the lectures. So after class today, I will be releasing it. And for about 24 hours, you will have access to that exam, giving you a chance to look at it, see what you got right, see what you got wrong, read the comments that I've made on your exams to help you to better prepare for the next ones or to continue to prepare the way you were. These exams will be available over the next 24 hours, so I expect you to use that time now, especially with the fast turnaround before our next exam, to look at those, to contact me if you have any questions. If for some reason you're not able to review it in the next 24 hours, after I close the exams tomorrow, exams will be available. If you want to come to my office hours, we can go over your exams there as well. So you will have another opportunity. If the next 24 hours is not convenient for you, then you'll have an opportunity to be able to do it outside of that time. And then, of course, obviously, if you have any questions, then definitely come to my office hours, speak with me, and I can help you to see what you got right and what you got wrong and what you can do to improve on that. All right. Any questions on that, this previous exam?
I have a question.
Yes.
So when you put in the grades, is the curve already applied?
Yes,
Or
these
okay.
grades are already curved. Yes, that is correct.
Okay. Okay, thank you.
Yeah, and in fact, when you look at your exam, at the very bottom of your exam, the way Proctorio handles the curve is that there is what they call fudge points. So at the bottom, you'll see the fudge points that were added into your score, which is basically how we adjusted the curve. So yes, so these grades are already curved. All right. All right, and again, you see that with the averages. There wasn't a person in the class who only got one question wrong on the lab exam. There wasn't a person who only got half a point wrong on the lecture exam. Those are the grades after the curve. So again, we had some people who did very well, but not quite that well. Everybody took advantage of the curves, and the curves were fairly generous. All right, any other questions on the exam stuff? All right, then let's talk about our game plan because it's going fast. Over the next week, we have a ton of material to cover. Today, we are going to finish our discussion of tissues. We have one more thing to talk about for epithelial tissues, and then we'll move on to our connective tissues. And then as we talked about, hinted at in the last class, we'll put those epithelial tissues and connective tissues together to make membranes. And then after that, we will start our introduction into the integumentary system, talking about the skin, finally getting to that level of our first organ system. If there's time, we'll continue our discussion of histology. Although, as I mentioned in the last class, I think the connective tissues are far, far easier to identify and distinguish than the epithelial tissues are. So we spent a little extra time on the epithelial tissues last time because I think that was the harder part. But if we have time at the end, we'll spend a little time on connective tissues doing that. And if not, then we'll save it for our review on Wednesday. Speaking of Wednesday, you have 3 assignments due: your Unit 6 review, your epithelial tissue and connective tissue handouts. Remember, not only are those your study guide for the tissue component of this lab exam, but they are a graded assignment. Remember, you're not being graded on your drawing skills, but I expect you to put time and effort into them. And I also expect you to label them. Remember, you have all those names of all the things I'm going to be testing you on. So label those pictures. And then, like I said, there's that fun fingerprint activity that you'll find in our assignments as well, all leading up to 1 week from today when we have our second lab and lecture exam. All right. Questions on any of that? All right, excellent. So when last we left off, we had talked about the structural classifications of our exocrine glands. And someone remind me again how many structural classifications there are?
8.
Yeah. 8 total. 1 is, of course, the unicellular. And then we saw 7 multicellular classifications. And we talked about how they're assigned based on their secretory structure shape, and then the number of secretory structures and the number of ducts. But as we also talked about in the last class, when identifying glands, there are 3 ways we identify the glands. We identify the glands by the substance they produce. So sweat glands produce sweat, right? Saliva glands produce saliva. The structural classification. And then the last thing we needed to do was the functional classification. Now, the good news for this one is there are only 3 structural classifications, and that's tiny. So that should make it a little bit easier. And your book here has got this simple illustration that does a good job of identifying those three types. But let's talk about and identify all three of these types, starting with this pretty picture right here. The first functional classification is the Holocrine mode of secretion. And that's the other thing to remember when we're talking about functional classifications. When we're talking about functional classification, we are talking about their mode of secretion. Or that's just a fancy way of saying how they release their substance. So let's take a look at what's going on in this cell that uses the Holocrine mode of secretion. Notice we start with information in the nucleus. As we now know, transcription takes place, making a messenger RNA that goes to the rough endoplasmic reticulum, where then translation takes place, making a sequence of amino acids, a polypeptide. That polypeptide goes to the Golgi apparatus, where it is folded, it is cut, it is modified, it is put together with subunits, and it is turned into a functional protein. That functional protein is then bundled up into a vesicle, and as you can see, that vesicle is brought to the surface of the plasma membrane, where the vesicle binds with the surface of the plasma membrane and releases its contents. So with the Holocrine mode of secretion, how does our cell release its contents? What is that process I just described?
Is it exocytosis?
Yeah, exactly, exocytosis. So basically, Holocrine glands basically use exocytosis. Now, does this modify or change or damage the cell? I hear your hesitation because it's kind of a silly question. What's the obvious answer here? Does it damage the cell?
No.
No, of course not. The cell is not changed by this process. It's a continuous process that this cell is able to do, and it doesn't affect or damage the cell in any way. This is by far the most common type that is found in the human body, the Holocrine mode of secretion. Here we saw what was happening in a single cell. Here is the nice picture from your textbook that does a good job of showing this. Notice again, we have cells that are making vesicles, bringing those to the surface, releasing those into the lumen, and then out the duct it goes. Because again, remember, all of our exocrine glands have ducts that carry it out of the body. And like I said, it's by far the most common type. Our salivary glands, our sweat glands, our pancreatic glands, dozens of exocrine glands in the body use the Holocrine mode of secretion. All right. Questions on this one? All right, let's look at the second. Oops, hold on. Let's do this. I want to leave that mode of secretion thing on here. All right. Let's notice what's happening here. Notice it is a similar starting process. We have transcription that takes place in the nucleus, translation that takes place in the rough endoplasmic reticulum, modified and cutting the protein in the Golgi apparatus, putting those proteins into vesicles. But notice this time, the vesicles aren't exocytosing. Instead, the vesicles accumulate in the apical portion of the cell. And then as it accumulates enough of this, what happens to the apical portion of the cell?
It detaches.
Yeah, the apical portion ends up pinching off. When it does that, it releases our secretion and some of the cytoplasm. And this is now way too big, not all going to fit in here. And some of the cytoplasm. This typically makes it a more viscous, a more organic type of secretion. And let's ask that question I asked before. Here, is the cell damaged?
No.
If I pinch off your arm, you're not going to be damaged by that?
Yes.
Yeah.
Yeah, absolutely, absolutely. Yes, the cell is damaged by this process. Does the cell die as a result of that damage? No, it's pretty much just a little bit of the cytoplasm and the secretions that it uses. It is able to recover and regrow. And as it recovers and regoes, it's going to fill up with secretions again. And then the process continues.
So would that be like dead skin flaking off, or is it different than that?
Different than that, because remember, when dead skin flakes off, the entire cell is being left, right? That is the protective measure of the skin. What we're talking about here are cells that are making glandular secretions. We have glands in our skin, but you wouldn't say that the superficial cells of our skin are glandular secretions. But that is the problem with this one, right? There are no human examples. So I can't give you a good example of what the epicrine mode of secretion is in a human. The best example that I can describe is actually outside of humans. And these are the anal glands of dogs. You may not often think of the anal glands of dogs, but even if you don't own a dog, you probably know it's the first thing a dog usually does when it meets another dog.
Smells their anal area.
Well, you can say but. This is an anatomy and physiology class. It's okay to say but. Absolutely. The dog will smell the other dog's butt. And why is he doing that? He is taking a whiff of those anal gland secretions, because those anal gland secretions, because they have that more organic component to them, tell them a tremendous amount about the health, the diet, the location where that dog has been, and all this other tremendous amount of information from that very organic release. Now, as I mentioned, this is a more organic type of secretion. So there have been a couple times where we thought there was a human example, and where you see this is in the skin. As you are aware, because we've talked about it since really the first day of class, if we were to all get up and run around the room 16 times, that would upset our homeostatic balance. And as a result of that, we would produce sweat to be able to lower our body temperature. However, at the same time, if I then take my gym shirt and I throw it in my gym bag and I leave it in the back of my car for 3 days, when I open that gym bag 3 days later, what's that experience like? Is that a pleasant experience?
No.
No, absolutely. It smells bad. And the reason it smells bad is it turns out we actually have two types of sweat glands. We have the watery sweat glands, like the ones that are on our forearm here, that produce that watery sweat to help us to regulate body temperature. But we also have these more organic sweat glands. We'll talk about this when we get to the integumentary system, but they're basically the ones that are found in areas associated with sexual maturity, right? My daughter, when she was 5, could run around the house for hours and still smell like a rose. However, now she's a teenager. And if I were to go into her bedroom right now, would it smell like a rose in there?
Probably not.
Probably not, exactly. Otherwise, I wouldn't be using it as an example. Absolutely, right? We have these secondary sweat glands. Again, humans don't have hormones per se, but it does produce a more organic sweat, a musky aroma that actually has a pleasant, most people find a pleasant aroma to it. Now, again, opening that gym bag in 3 days certainly isn't something that's pleasant, but that's because over those 3 days, bacteria have been feeding on that organic sweat in there, and it's that bacterial release that causes that problem, that aroma. And so here's the thing. When they first found these sweat glands in humans, they thought, these are producing a more organic sweat. So these organic sweat glands must use the Holocrine mode of secretion. So they called them Holocrine glands. And in fact, when we get to the integumentary system, we won't talk about it till Wednesday, but on Wednesday, we will talk about the sweat glands and we will talk about Holocrine glands. And they're still called Holocrine glands. But here's the problem. After they named them Holocrine glands, they continued to study them and they found out that nope, they actually release their contents by exocytosis. So what is the functional classification of our Holocrine glands? What was the mode of secretion where we use exocytosis? Do I need to flip back the slide so that you guys can remember?
American.
There you go.
American glands?
Exactly. So when we get to the integumentary system, we have glands that are called Holocrine glands by name, but do they actually use the Holocrine mode of secretion?
No.
No. So why didn't they change their name?
Anatomists hate us.
Exactly, because anatomists hate you. I don't have another good reason why they wouldn't. That's the only reason that they wouldn't. So does that mean there's absolutely positively no examples? Well, maybe. There is the possibility that the mammary glands or some of the mammary glands may use the Holocrine mode of secretion. In fact, if we went back to that original picture that showed the 3 types, they showed a mammary gland. As those of you who have ever breastfed or been breastfed may know, although you probably didn't know at the time you were a baby, there are basically 2 types of milk. There is a fore milk, which is more watery and more sugary, and there is a hind milk, which is a little bit more viscous and has a lot more lipids in it, for instance, and proteins in them. And so because there's those 2 different types of milk, there are some anatomists who believe that there may be 2 different types of mammary glands producing those 2 different types of milk. And so there may be some glands in the mammary glands that use the Holocrine mode of secretion. This is something they're continuing to study, and they're not wanting to jump the gun like they did with the skin. So they have not said with certainty. They say they believe that it may be the mammary glands, but we don't know that for certain. And since we don't know that for certain, for right now, we'll stick with the company line that there are no confirmed human examples. So there's maybe the mammary glands, but there's no confirmed human examples of that. All right. So obviously, we need to know this is one of our 3 functional classifications, for instance, for an essay question, but obviously there isn't a human example we could use, but luckily there is an animal example we could use if we needed to describe that. And does that sound like a possible essay question? Describe the functional classifications of exocrine glands and give an example of each. Yeah, that sounds like it could be an essay question to me too. All right. Let's take a look at our last mode of secretion. The last mode of secretion is the holocrine gland. I love this particular picture. This one's not from your textbook, but it amuses me, which is why I use it. What process is going on right here?
Oh, sorry. Yeah, telophase.
Is it? Oh, I can understand why you would say that. However, it is indeed cell division. We are seeing the separation of the chromosomes, and this was our artist's representation of anaphase. However, if you notice for anaphase, they have their V's pointing in the wrong direction, right? So again, you got to love those artists and that artistic license that they get. But what we have here are cells that are dividing, producing new cells. Those new cells are filling with the secretion. And when they get to the top, these cells basically burst, releasing all of their contents. That means not only are they releasing the secretion, and all of the cytoplasm. This produces a very thick, a very viscous, very organic secretion. And let's ask that silly question we've asked twice already. Does this damage the cell?
No.
Yes.
Or yes.
Yeah, having the cell rupture and release all of its
Rupture and
contents.
release. Yeah.
Popping a balloon is definitely something that damages it. In fact, not only does it damage it, but the cell actually dies in this process, right? But the process can continue because we keep making more cells. Again, this is a very simple illustration. Here's the nice one from your textbook. Notice again, here they've got the V's pointing in the right direction for our mitosis that is taking place. We have our cells accumulating, filling with the substance, and then just rupturing and bursting, releasing that big, thick, viscous, organic secretion. This process is not nearly as common, but it is used in, for instance, our sebaceous glands of the skin. Sebaceous glands of the skin are what we commonly refer to as the oil glands of the skin. The sebaceous glands are the ones that become more active in the presence of hormones, like those puberty hormones. So one of the things that starts to happen as we age, as we enter sexual maturity, is we get an increase in production of this substance. And as we're increasing production of it, it's actually possible that this duct can get congested and blocked with this thick organic material, which, as we mentioned, is a perfect breeding ground for bacteria. So bacteria can get in here. This area can become enlarged and inflamed. And what do we call that condition when it occurs?
Acne.
Yeah, acne, absolutely. That acne that forms is from the blocking and congesting of these holocrine oil glands, producing that thick, viscous oil on the surface of our skin. But like I said, if they become overproductive or they become productive or become congested, then that can lead to bacterial infection and inflammation, redness, swelling, and that's our acne.
You said this was anaphase happening?
No, the illustration, what I was saying is the illustration is showing anaphase. What is happening here is mitotic division, right? We are having the cell dividing, and as these cells divide, they push the old cells towards the lumen. As they reach the lumen of our duct, they rupture and burst, releasing all their contents. So as we mentioned, if the cells die, we have to be able to make more cells to continue this process. So basically, there's really three things that happens. Cells divide, cells fill with sebum. Sebum is the name for the oily substance that the cell produces. and then the cell ruptures, releasing everything, and dies. And the process continues. All right. Questions on that? So those are our three. Let's write this again just to remind us. 3 functional classifications, or remember the other fancy way we could say this is mode of secretion. Right? How it is that it releases its substance. And there are 3 of those.
I have a
All
question.
right. Yes.
So we need to know what each gland produces. And so for the apocrine glands, what exactly do they produce?
So
Again?
let me stop you right there. For the focus of what we've been talking about right now has been just to talk about the general shapes and the general way that they release their contents. Now,
Right.
for each of those, we did give an example because, as we mentioned, those make great essay questions. Identify the structural classifications of exocrine glands, the functional classifications of exocrine glands, give an example. So those definitely are things that you need to know for the exams information. However, from now till the end of 431, every time we run into a gland, there are going to be 3 things about that gland you're going to need to know. You are going to need to know the substance it produces, the structural classification, and the mode of secretion. In fact, we already have learned in this class 1 gland. That 1 gland we've already learned is the submandibular gland. And hopefully, since it's the only gland we have really learned about so far, give me those 3 pieces of information. What is the substance it produces? We learned about the submandibular gland at the end of our last class. What was the substance it produced? There we go. Mitch has got it. Saliva. Excellent. What did we say its structural classification was? So glad this is information that probably won't be on the exam. Oh yeah, it absolutely will. What was the structural classification of the submandibular gland? It's on your epithelial tissue study guide if you don't remember. We talked about it at the end of last class. It made both serous fluid and mucus. So it has 2 different types of secretory structures. So what must its structural classification be?
That's the tubulo-alveolar.
Exactly. Compound tubulo, alveolar. Excellent. And after we talked about today, what do you think its functional classification is? Holocrine, exactly. Spectacular. Thank you, Sarah. Excellent. So there you go. So we haven't talked about the glands of the skin yet, but we will be doing that on Wednesday. So when we talk about those glands of the skin, we will absolutely need to know what they produce, what their structural classification is, and what their functional classification is. So we will get there. All right. Excellent. Any other questions on our epithelial tissues before we move forward? All right, perfect. Then let's talk about our connective tissues. Like I said, the good news is I think the connective tissues are a little easier. They're the most abundant and most widely distributed of all the tissues. They are also the most diverse. and both diverse in structure and also in function. So they look very different from each other, and they also function very differently from each other. Give me some examples of some connective tissues that you know of.
Loose connective.
Say again?
Loose connective tissue.
Excellent, absolutely. Loose connective tissue. Where would you find that in your body?
In your organs.
Where?
Your organs. Lining.
In your organs. How about fat? Fat's an example of a loose connective tissue. Your subcutaneous fat, that's an example. Give me another example.
Bone tissue,
Say again?
like the in your bones.
Yeah, bone. Bone connective tissue. Excellent. All right. Give me one more.
Nervous tissue?
Well, remember, nervous is its own tissue type. It's not a connective tissue. But what about blood? Blood is actually a connective tissue. Think of those three things: bone, blood, and fat. Right? Those are three very, very different substances, and yet they are all connective tissues. Very different structures, very different functions. As we talked about, obviously, with a name like connective tissue, their job is to hold things together. They provide protection, they provide support, they hold things together. But bone provides the ability for movement, right? Our blood is involved in transportation and our immune response. Adipose stores energy. I mean, there's all these different things that are going on with them. Now, if you remember, when we talked about epithelial tissues, there were some characteristics that we talked about that all of them shared. Remember, we talked about how they're all superficial, they're all polar, they're all very cell dense. So there are characteristics that all epithelial tissues shared. When we talk about things like bone and blood and fat, those things are so different from each other. Are there things that they're going to share? Well, it turns out yes. Maybe not a ton of them, but absolutely there are some characteristics that they share. For instance, all connective tissues are deep tissues. What does that mean?
Away from the surface, or
Away from the surface, right? Remember, one of the keys with epithelial tissues is epithelial tissues all have free space above them because they're lining surfaces. Does your connective tissue have free space above it? No. If your connective tissue is showing, go see a doctor because it's not supposed to be showing. It is a deep tissue. It does not have a free surface. It's also not polar. Remember, as we talked about, if I were to take my skin and flip it upside down, would it function the same way?
No.
No, but if I took a drop of blood out of my blood vessel, rotated it 90 degrees, and then put that drop of blood back into my blood vessel, would that change the way that my blood functioned in my body?
No.
Or if I took a little cube of my fat and rotated it, or if I took a little cube of my bone and rotated it, no, that's not going to have effect. So there's no polarity to it. It's not polar in the same way that our epithelial tissues are. Like epithelial tissues, they are innervated. They have nerves that go into them, and we do receive information from them. Remember, there's a large amount in our epithelial tissues. There's a fair amount in our connective tissues. But remember, our epithelial tissues were avascular. They had no blood supply. Whereas our connective tissues typically are very well vascularized. Now, of course, what's the key word in that sentence?
typically,
Typically, because what does typically mean?
not always.
Not always, absolutely. Why might the amount of blood matter? Why might how much blood goes to a connective tissue make a difference?
Blood pressure?
Okay, some of it's going to be blood pressure, but that doesn't really affect the tissue itself as much. Why does it matter to the tissues? There we go. Mitch has got a good idea. Healing, right? It's that magical time of year, right, when everybody gets super excited about fantasy football. And if your first round draft pick, that running back, ends up breaking the femur in his leg during the first game of the season, you're pretty bummed about that. And how long is he out for? You break a bone. How long does it take that bone to heal?
Month.
Yeah, month, 6 to 8 weeks if it's a bad break, something like that. 4 to 8 weeks, right? Biggest bone in the body, and they could be out as little as 4 to 8 weeks. Why? Well, because bone has a huge blood supply to it. But what if instead he tears his Achilles tendon? How long is he out for then?
Not longer.
Yeah, much longer. It can take a year, in some cases 2, for a tendon or a ligament to heal. Ligaments and tendons are poorly vascularized. They have a slow blood supply to them. And what if instead he tears the cartilage in his knee? How long is he out for then?
Forever.
Yeah, in that case, yeah, Crystal, you guys got it right. Absolutely, forever. At that point, does that cartilage in the knee ever really heal properly? No, cartilage is avascular and it heals incredibly slowly. And so his football career may be over as a result of it. So notice most connective tissues have a very large blood supply. And as we just finished talking about, that can have a huge effect on how rapidly they heal. All right, excellent. So those are some characteristics that all connective tissues share. But let's talk about some other ones. The other big difference between connective tissues and epithelial tissues are connective tissues are more cell sparse. Remember, with epithelial tissues, we said epithelial tissues are cell dense, basically made up of many layers of cells smacked together, and there's not a lot of room for other stuff. But connective tissues have fewer cells. And so that means there's more room for more stuff. So when we make a connective tissue, absolutely, we have to have cells because after all tissues are made of cells. But the other big key to a connective tissue is a connective tissue is going to contain many fibers inside of it. And it also is going to, all right, I'm getting feedback from someone, so I'm going to mute you guys all, but again, you're always welcome to unmute, ask questions if need be. But also, it is going to have this fancy stuff called ground substance. In simple terms, and we'll talk about it in this a little bit more in just a minute, ground substance is basically water and the stuff that is dissolved in the water. It's a little bit more sophisticated with that, but that's a good starting point for what that is. So we have cells, but we also have fibers and we also have ground substance. And that fiber and ground substance together forms what we call the inorganic matrix. And the matrix determines the shape and the function of the connective tissue, right? For bone, the matrix is basically calcium salt crystals, which makes bone strong and hard, right, and sturdy. For blood, the matrix is the plasma, which is mostly water, making it a fluid, right? There probably aren't two different substances in the body than bone and blood, but both of them are connective tissues, and it is their matrix that determines their shape and therefore also their function. This matrix is really the cardinal way we define connective tissues. Right? If you think of epithelial tissues, we said epithelial tissues were on the surface and they're cell dense. That was kind of their cardinal characteristics. And connective tissues, the cardinal characteristic is they have this inorganic matrix, which pretty much tells us everything about it. Now, of course, it is a, around time, doing good. It is a tissue. It is made up of cells. And so obviously we have to talk about the cells. And all cells of connective tissues are formed by the same pluripotent stem cell. Many of you got the essay question involving stem cells on this last exam. Someone remind me again what a pluripotent stem cell is?
A stem cell that makes like any type of cell or
Any?
produces or
No, it's only like two or three types.
Sure, well, a small amount.
It's a
absolutely.
section, right?
These are ones that can differentiate into multiple cells, but not everything. Remember, the everything ones are the omnipotent.
oh yeah. Okay.
Yeah.
That's-
Unipotents are one thing. So basically, pluripotent is somewhere in between. So every single cell in a connective tissue comes from one pluripotent stem cell. And that pluripotent stem cell is a mesenchymal cell. Now, when we make the cells for our connective tissue, they basically come in two flavors. They're the immature cells, which are the blasts, and blasts are basically responsible for making the matrix. And the mature cells are the sites, and their job is to maintain the matrix. Which of these two cells do you think are more active, the immature cells or the mature cells?
Mature.
Well, which do you think it's harder to do? Do you think it's harder to make the matrix, or do you think it's harder to maintain the matrix?
Making.
Yeah, making. And again, who has more energy to run around the house? Your toddler or grandma? Right?
Toddler.
Yeah, exactly. So these are the more active cells. Making the matrix is a lot more work than just maintaining the matrix is. So when we have our cells of the connective tissue, we have things like fibroblasts and fibrocytes. And guess what fibroblasts make and fibrocytes maintain?
Fiber.
Yeah, they make the fibers, absolutely. Chondrocytes and chondroblasts. What do those make and maintain?
The cartilage or
Cartilage, excellent. Osteoblasts and osteocytes. What does osteo refer to?
Bone.
Bone, absolutely, right? Adipocytes are the fat cells. And again, as we know, our anatomists love to give us, curveballs. So while we have things like lymphocytes, we also have things like mast cells and macrophages. So this blast and site rule isn't followed 100%, but many of the cells of our connective tissues are either going to be blasts, the immature cells that are making the matrix, or the sites that are going to maintain them. All righty, those are the cells. So let's talk about the fibers. Fibers are what provide the strength and support for our connective tissues. And there are three types of fibers. The first type of fiber is a collagen fiber. It is made up of the protein collagen. In fact, collagen protein is the most common Protein in the body. So if you were to randomly reach into the body and pull out a protein, it most likely would be a collagen protein. Collagen fibers provide tensile strength. Anybody know what that means?
Really strong.
Yes, but in particular, it is really strong in resisting tearing, resisting shearing forces, right? If I were to divide the half in class, and again, I have to work on my analogies because we're not in the classroom, but I'll pretend that we're all in the classroom. If I were to divide the classroom in half where we're all together, put half of you on one side of the room, half of us on the other side of the room, throw a rope into the middle, and we spent the next remaining 3 hours of the class playing tug of war, as big and strong and fit and healthy as all of you are, with half of you pulling on one side of a rope and half of you pulling on the other side of the rope, are you going to be able to pull that rope in half, tear that rope into 2 pieces?
No.
No, of course not, right? Because it has tremendous tensile strength. It is strong. It doesn't break. But notice that strength doesn't come from it being rigid, right? When we're done playing tug of war, I can roll it up into a ball and I can toss it into the trunk of my car. So that strength doesn't come from being rigid. It just has all those fibers that resist that shearing, tearing forces. And that's why we have so much collagen. It's in our skin, so that when I pinch your skin and pull on it, that skin doesn't come tearing off, right? Again, you may not have thought of it in those terms, but if you don't have lips like Angelina Jolie, what can you do?
Go get filler.
Yeah, and what is filler?
The collagen protein.
Collagen. Yeah, they put the collagen protein in there. It gives it structure and support without making it rigid. Right? So if you want lips like Angelina Jolie, a butt like Kim Kardashian, right, we can inject collagen into those areas and it'll give that strength, that support, that firmness without rigidity. All right. However, there's something else that happens with my skin. When I pull on my skin, it doesn't tear, but when I let go, it goes back to its original shape, right? I can grab my kids by the scruffs of their necks and carry them around the house. And when I let go, the skin goes back into place by the time Child Protective Services get there, right? And that's because skin has elasticity. And that elasticity comes from elastic fibers. Elastic fibers are made from a special elastic protein called elastin. And it has that stretch and that strength, but it also has that ability to recoil, just like a rubber band. If I take a rubber band and stretch it out and I let go, what happens?
Snaps back.
Yeah, it goes back to its original shape. Now, are there limits?
Yes.
Yeah, if I stretch that rubber band too far, what's going to happen?
Chub.
Yeah, it's gonna break. It's gonna
Break.
tear.
Yeah.
Absolutely, right? And is the same thing true for your skin?
Yes.
If you reach that magical time in your life where you decide that over the next 40 weeks you want to grow a basketball inside of you, right? You have that ability to expand the belly tremendously as a result of that. But can that rapid growth and expansion put too much strain on the skin and cause stretching to occur?
Yes.
Right? And if it stretches too far, what happens?
Stretch marks.
Stretch marks. And it's not just pregnancy that can do that. My wife's brother, his senior year in high school, grew 12 inches. Right? She went away to college. She came back at the end of the year and he had grown literally a foot in height. And that much rapid growth was too much strain and stress. And so he has, or had, because it's been many years now, huge stretch marks on his side from that incredibly rapid growth, stretching the skin beyond its elasticity.
So obviously people have different amounts of elasticity in their skin because some women don't get stretch marks, some women do.
Yes. Absolutely. And the amount of elasticity tends to be genetic. So again, if your mother had stretch marks, then the female is more likely to have stretch marks. I know there's all sorts of lotions and other things that you can, put on the skin to supposedly help with that, but really it doesn't. There's not much that can be done. It's really just a matter of how rapidly the body grows and how elastic it is.
Yeah, I'm 6 months pregnant, so nothing yet.
Congratulations. Just the first?
Yes.
Oh, very cool. Excellent.
Nothing else either.
I'm sorry?
Same here, no stretch marks.
Oh, excellent. Congratulations. to both
Crossing
of
my
you.
fingers. Crossing my
See,
fingers.
normally I would give you my speech about not having kids and how horrible kids are, but since it's too late for you guys, I'll spare you that
Yeah.
that speech.
Please do.
All right, excellent. So that's our second type. There is one third type of fiber in our connective tissue, and it is called a reticular fiber. Now, one of the interesting things about reticular fibers are also made of the collagen protein. The difference is the arrangement. In collagen fibers, our collagen proteins, much like that rope we talked about, are long threads of collagen proteins that really do form like a rope or a cable-like structure, which again gives that resistance to the tearing. With reticular fibers, these fibers are short and elaborately branched. So it's still the collagen protein, but the fibers are much shorter, much more elaborately branched. And what they do is they produce a very porous, a very open, a very soft and spongy type of tissue. So this is like our bone marrow. This is found in lymph nodes. This is found in the liver and the spleen, things along those lines. If you've ever cooked liver, when you've gotten that liver, it is very spongy, much more spongy than, for instance, that, that steak that you're gonna cook instead. So it's because it has this spongy reticular fibers with all of this space for cells and other stuff inside of it. And like I said, we'll see some of these examples of these fibers when we look at them under the microscope, when we look at our connective tissues. All right. So question, 3 types of fibers made up of 2 types of proteins. Questions on any of that?
Can you give some examples of reticular fibers?
So yes, like I said, in the lymph node, in our liver, and our spleen, bone marrow, all of those are examples of places where you find reticular fibers that are used to make reticular connective tissues. And there's other places we'll find them as well. We'll look at it under the microscope and see it. All right. Any other questions on our fiber types? As I mentioned, the second part of our matrix, the third component of a connective tissue, is our ground substance. And as I mentioned, our ground substance is mostly water and the stuff dissolved in the water. Its job is basically to help to hold the cells and the fibers together. And because it's water, that is where the chemical reactions are going to take place. That's where the exchange, where the tissue is able to get the oxygen and give off the carbon dioxide, get the nutrients and give off the wastes. So it's also going to be where all of the medium of exchange is going to take place. As I mentioned, your book's got a great table that talks about all of the stuff that can be in the ground substance. I think it's pointless to just sit there and rattle them all off, but I do want to talk a little bit about some of the stuff. That obviously, like I said, this is the interstitial fluid, which, as we know, basically interchanges with the blood plasma to help to regulate what's going on in our cells. Again, Our job is to hold the tissues together. So there's going to be a large amount of polysaccharides, making the tissue sticky, helping to hold it together. And since our body is pretty much 50% of the stuff of our body, let's say it that way, is proteins, not surprisingly, there's a massive amount of proteins in there as well. Again, I could spend the rest of the class time just listing off the proteins in our tissues. However, there is one I want to talk about. Because remember, we talked about bone, we talked about fat, but we also talked about blood being a connective tissue. And if you had a bucket of fresh blood and you were to reach your hand in there and pull it out, would you find any fibers inside of that blood?
No.
No. So it doesn't sound like it would meet our criteria. But it turns out blood has a couple, well, it has several different, but the two most common are fibronectin. Oops. And fibrinogen, fibrinogen, which are two very special protein fibers because they are water soluble. These are fibers that are actually able to dissolve in water, so that when you reach in and pull your hands out, you don't find a bunch of fibers in there. However, if you cut your arm and blood is coming out as a result of that, don't we want to stop that blood loss?
Yes.
Yeah, and that clot that forms is caused by bringing those fibers out of solution. When we bring those fibers out of solution, we form this big mesh net that forms the clot and then will harden, dry and harden to become the scab. So that clot, that scab that forms is by actually bringing those fibers out of solution. So when you look and you give that pint of blood at the Red Cross and you look in the bag, you don't see any fibers, but there are indeed some fibers in there. Blood definitely is a connective tissue. And so these are a couple really interesting ones because of that unique characteristic of dissolving in the water. But like I said, we could basically spend the rest of the class time just lifting off proteins that are in the ground substance, which wouldn't be tremendously fun.
So what makes blood a connective tissue is the fibrinogen or
Yes,
Fibronectin?
fibrinogen and fibronectin, because again, remember, to be a connective tissue, you have to have cells, you have to have ground substance, and you have to have fibers. And so because we have several different types of water soluble fibers, blood is indeed a connective tissue. Now, as last thing that we'll talk about before we take our first break, as I mentioned, unlike epithelial tissues, which we can identify by the shape of their cells and the number of layers of the cells, there really isn't a good way to distinguish the different types of connective tissues. So the rigidity of the tissue is as good as any other characteristic. Obviously, as we've talked about, bone is by far the hardest of our connective tissues, followed shortly by cartilage. This is then followed by our dense connective tissues and our loose connective tissues. Dense connective tissues are also sometimes referred to as fibrous connective tissues, because guess what a dense connective tissue is densely packed with?
fibers.
Yeah. So that's where it gets its name. And then loose connective tissues have fewer fibers and more cells. And then, of course, last, our softest of all of our tissues, blood. So again, there's no hard and fast rules. This is as good as anything else, but it gives us our 5 main types of connective tissues. So these are our, let's actually say it this way, 5 general types. Kind of like we had simple epithelial tissues and stratified epithelial tissues. But didn't we have multiple types of simple and multiple types of stratified?
Yes.
Yeah, and the same thing's going to be true here. We have two specific types of bone. We are going to have three specific types of cartilage, three specific types of dense, three specific types of loose, and blood is just blood. So after the break, we'll come back and talk about the specific types of bone and the specific types of cartilage. And just like we did for the epithelial tissues, we'll talk about their characteristics. We'll talk about how we're going to recognize them on the microscope, and we'll talk about where we would find them in the body. All right. Questions on that? All right, let's go ahead and take our first break. Looks like it's 9:05 now, so we will restart at 9:20. Take a good 15-minute break, and I will start the recording at that time. All right, any questions before we take our first break? All righty, see you in 15 minutes. So let's take a look at our connective tissues. As I mentioned, bone is our hardest of our connective tissues. And again, remember, we are talking about bone connective tissue. I think some of the confusion that comes in with this involves the difference between bone connective tissue versus bones that are organs. The humerus, the femur, right? The bones of the skull, those are all bones. Those are things that are organs. However, obviously, the primary tissue that makes our bones is bone connective tissue. So bones and bone connective tissue are not interchangeable terms. I appreciate that can be a little confusing. If it helps, you are welcome instead to use the term osseous tissue instead of bone connective tissue. They basically both mean the same thing. And remember, as I also mentioned, there are two specific types. And those are compact bone and spongy bone. Now, obviously, the very next section of this class is going to be the skeletal system. So we will talk about these in much more detail then. So for this exam and for this exam only, we are going to focus solely on compact bone. So we won't worry about spongy bone yet. We'll talk about that more when we get to the skeletal system in the next section, which is going to be next week. And compact bone is what you think of when you look at that skeleton. When you look at that skeleton and you see that white uniform outer surface, that is the compact bone. Now, while it looks white and smooth and uniform on its outer surface, when we look at it under the microscope, we can see that there is a whole lot going on. And one of the keys to our compact bone is, if you notice, compact bone is primarily made of these circular shaped structures. Of course, as we know, anatomists love to name everything. And so not surprisingly, we have names here as well. Compact bone is made of, or let's say it this way, the organizing structure is what we call an osteon. So an osteon is this round structure. Notice when we look at the osteon, it has a space at the center. And not surprisingly, that space at the center is called the central canal. This is all too big. Any idea what that central canal might contain?
a nucleus or something?
It's a great guess. Normally when we have spaces like that, that might be a cell in there and a nucleus in there. And there are indeed cells inside of this osteon, but not in the central canal.
It's not a goblet.
Nope, there are no cells. It's struck. This is a big enough
This is
hole where there are
structure.
actually structures in here.
Okay. Artifacts.
Remember, artifact is something artificial that shows up when we make it. Although what we have is similar in there about arteries,
Oh.
blood vessels. Remember, one of the things we talked about, bone is a very dynamic tissue. It heals very rapidly. And that is because there's a large blood supply. We talked about depending on the type of break, depending on the bone, it can heal in as little as 4 weeks. Does that mean you should go running out and break a bone as every day, 2 on Sundays?
No.
Why not? Because what else passes through these central canals? Well, is breaking a bone a fun experience?
It has nerves.
Nerves, yeah.
There you go. Nerves, absolutely. So blood vessels and nerves pass through that central canal. But you are right, we need cells. Now notice this is a big solid matrix. So what we're going to need are mature cells that are going to maintain this matrix. And what would the mature cell be that maintains the matrix? Maintains bone.
Osteocytes.
Excellent. We would need osteocytes to be able to maintain this solid matrix. And notice it is a solid matrix. Okay. So if our osteocyte is going to be able to live in this matrix, that osteocyte needs a space to live, right? If a bear lives in a mountain, that bear needs a cave, needs a hole inside of there where it can live. And the same thing is true here. Notice all of these black dashes that we see all over the place. These are the caves that our osteocytes are going to live in. And of course, as we know, anatomists love to name everything. So our osteocytes live in the space called a lacuna. Lacuna being the singular, the plural, lacunae. Oops. With an E. So with an E or without an E, singular and plural. So bears live in caves, osteocytes live in lacunas. All right, questions on that? Anybody have their histology list handy nearby? Is there anything else we need to know for our bone connective tissue on that histology list? Osteons, central canals, osteocytes, lacunas. Are you guys silent because you don't have your histology list, or are you silent because I've named everything on it?
No, I think you got everything.
Okay, excellent. Now, notice there is one trick on this though. Notice, do you actually see any of the osteocytes? Do you actually see a cell that has a nucleus or cytoplasm or anything like that? No, Jackie doesn't see it, and I don't see one either. So notice on the exam, what I would do is I would have an arrow like this, and notice I could ask one of 2 questions. I could ask you to identify the cell that would be located here. And if I asked that, what would you say?
Osteocyte.
Right? Or I could say, identify the space at the tip of the pointer. And what would you say to that?
Lacuna.
There you go, exactly. So notice again, that's how, so even though we really can't see the osteocyte, we know it's in there. We know that bear's living in that cave. So again, I could either ask for the cell or I could ask for the space and ask for those two different answers that way. All right, excellent. So we did all of that. Again, remember when it comes to the matrix, our matrix is basically calcium salt crystals. But there is some collagen fibers, giving it some flexibility, giving it some give, right? Probably the best example of this is the difference between our bones and our teeth. Both our bones and our teeth have the same calcium salts that give them their hardness. However, our bone has collagen fibers that give them some flexibility, give them some give. The enamel of your teeth doesn't have those collagen fibers. So does the enamel of your teeth have that flexibility, have that give?
No.
No, right? So if you're eating that big old bowl of clam chowder and you bite on a clam shell, right? Does that enamel have flexibility? Does it have any give?
No.
No, it has strength, but it doesn't have those collagen fibers. It doesn't have that tensile strength. And so it can break, it can shatter as a result of that. Whereas our bones have a little bit more flexibility and give, which is why when you jump up and down, right, your bones don't snap because they have some flexibility and they have some give. In fact, when we're born, our bones have more flexibility, more give, right? If you think about it, you can hold a baby at chest height, bounce, drop that baby, and it'll bounce right back into your arms, right? We talked about that in the last section. All right, I know a couple of you are pregnant, don't drop babies, but there are plenty of babies that roll off of changing tables, and typically are they injured by that? No, because their bones have more flexibility and give. All right, so that collagen is an important part of the bone. Speaking of give, speaking of collagen, that takes us to our cartilage. Cartilage is similar to bone in that it is a solid matrix, right? But it is less hard than bone and has more flexibility. And that's because it doesn't have those calcium crystals in it, but it does have a massive amount of collagen. Now, because it's a solid matrix, it is a tissue, it needs cells, and it needs spaces for the cells to live. So what do you think we call the mature cell located inside of our cartilage tissue?
Chondrocyte.
Is the cell. And what do you think we call the space that it lives in? It is also a lacuna. Yep, exactly. Jack has got it. It is also a lacuna. So lacuna is the space that a cell lives in a solid matrix. So chondrocytes live in lacunas, osteocytes live in lacunas. Now notice, and again, I like these pictures from your textbook because they give us an artistic representation of it on one side, and then what it actually looks like under the microscope under the other. All cartilages have chondrocytes. All cartilages have lacunas. So the way we're going to tell them apart is by the matrix. What we're seeing here is the most common type of cartilage, what is known as hyaline cartilage. Hyaline cartilage, like I said, is the most common type. And the way we can identify it is that it has a clear matrix. Notice whether we're looking at the artist's representation or whether we're looking at the real tissue under the light microscope, we don't see any fibers. It is a smooth, uniform matrix. This gives this hyaline cartilage a nice, rigid, hard feel to it. Where might you find hyaline cartilage? Anyone know where you might find this?
Your ears?
Well, so joints, Mitch has got one. Joints is a good one. Not the ears, but the bridge of your nose. I don't mean the tip of your nose. The tip of your nose, that's all rubbery. That's a different type of connective tissue. I'm talking about the bridge. The bridge of your nose is hyaline cartilage at the end of your joints. Hopefully you haven't seen the end of your joints, but now that the world is opening up again, you have the opportunity to go out and do things. So one of the things that I'm sure you've wanted to go out and do is go to a Renaissance fair. And of course, the whole reason to go to a Renaissance fair is so you can get those massive turkey legs to be able to eat them. And while you're peeling all the meat off of the big thick end of the turkey leg, if you look at the other end of the turkey leg, there's that white rubbery material on the end. That's that hyaline cartilage at the end of the bones. So hyaline cartilage is found in the joints. It's found on the bridge of your nose. However, if you feel the bridge of your nose and feel your ear, do those two things feel the same?
No.
No. That's because your ear is made of a different type of cartilage. The cartilage it is indeed cartilage in your ear, but it is an elastic cartilage. Notice we again have lacunas. We again have chondrocytes inside those lacunas. But remember, as we talked about, look at the matrix. And notice here, the matrix is chaotic with all of these thin fibers in all directions. These thin fibers in all directions turn out to be elastic fibers. And elastic fibers are what give this cartilage its bendability, its flexibility that we have in our ears. Anywhere else we might have elastic cartilage? Well, I'm asking the question.
Skin?
Skin's not a bad guess. We have elastic fibers, remember, but not elastic cartilage. So there are elastic fibers in the skin, but not elastic cartilage.
Like our elbows?
So again, remember, as we talked about, joints are where we're going to find the hyaline cartilage because we want that flexibility, we want that give. But let's jump ahead, let's cheat. Here is, I don't want to have to erase all this, so let's just do that. Clear all that.
I have a question too, really quickly.
Yeah, go ahead.
So I noticed on the histology for the epithelial tissues, you asked for the specimen or location, but on the connective tissues, you didn't have that on there. So are we responsible for knowing where to find these tissues?
So remember, I won't ask the question specimen because if you look at a bit of elastic cartilage, for instance, you can't tell just by looking at that elastic cartilage where it's from. So I wouldn't ask the specimen question that way. But could I ask you for one location where you would find this tissue? Absolutely. So one of the places you'd find elastic cartilage would be the ear. The other place you would find it is actually this structure right here. This structure right here is what is known as the epiglottis. Okay. Put your hand on your throat and swallow. When you do that, you feel that bump, that Adam's apple moving up and down. That Adam's apple is one of the organs, which was actually on your lab exam, known as the larynx. The Adam's apple is an enlargement of the larynx, and in the larynx is a special structure called the epiglottis. When you swallow, notice the larynx moves up. When the larynx moves up, that causes this bendable, flexible epiglottis to, oops, hold on, let's change colors, to fold down over the opening to your airway so that your food and your drink go into the esophagus and don't go into your lungs. So that bendable epiglottis inside your larynx is one of the places, and hopefully you can't feel that. No, not hopefully you can't feel that, unless I guess you stick it way down your throat. But obvious place where you could find it is here in the ear. And here in the ear reminds us of another important, so undo, 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 undo. Perfect. Reminds us of another important piece of information that we learned about our cartilage. Notice in these pictures that we've been looking at so far, there are no blood vessels. Cartilage is avascular. And remember, as we talked about, that affects the way that it heals, right? Ears are a great place where you see that, right? If you wrestled in high school, one of the things that commonly happens is your ears get beat up and that cartilage can be broken. And does it necessarily heal right away?
No.
No, but they continue to wrestle. And as they continue to wrestle, they continue to break and re-break and damage that. And what can happen as a result of that?
Cauliflower ear or whatever.
Yeah, that cauliflower ear, right? They start to get this enlargement. And in some cases, it can actually swell up and become bluish and white and kind of fold over. And they get that, that little bit of cauliflower ear as a result of the damage to that cartilage. I wrestled in high school, which was 875 years ago, and I actually still have a little bit of that cauliflower ear on one side because it basically never heals properly because it doesn't have any blood supplied to it. All right. That brings us to our third type of cartilage, and that is fibrocartilage. Notice again, where we look is the matrix. And again, notice in the matrix, we again see fibers. But notice this time, the fibers are not dark. They're not distinct. They're typically lighter in color. And notice they're more parallel in their arrangement. And that's because unlike elastic cartilage, which has these thin, dark elastic fibers, fibrocartilage contains collagen fibers. And this parallel arrangement of this allows this tissue to resist compression. Basically, it acts as a shock absorber. Anyone driven in a car that has problems with the shock absorbers on them? Come on, usually there's at least one or two of you. Yeah, excellent. Yeah. Was that an enjoyable experience?
No, it bounces.
No.
Yeah, you feel every single tiny little bump and pebble on the road. Absolutely. So having that shock absorption is very, very important. Not surprisingly, one of the places where you find this fibrocartilage are in the discs between your vertebrae. Because every time you take a step, you don't want your brain rattling around inside of your skull. So it provides that compression. It provides that, shock absorption as we walk. Or, since a couple of you are pregnant, let's mention another place where you would find this. Oh, here we see both of them. Here, notice this is that intervertebral disc that is made of fibrocartilage. That blue doesn't show up well. Make it red. In between the vertebrae, helping that shock absorption. But notice also here on the front of the pelvis is a little chunk of fibrocartilage forming a special joint known as the pubic symphysis. Notice they have the Latin here. The pubic symphysis. The pubic symphysis helps to hold our pelvis together, providing our center of balance, our center of support. But as we've also talked about, Females get the honor and the privilege of passing a basketball through their pelvis. And so one of the things that typically happens during the late stage of pregnancy is the female hormones cause a loosening of that pubic symphysis, making it more flexible, giving it more give, making it easier to pass the baby out. You may not have thought of it in these terms, but I know you're aware of it, because often when a female is in late stage of pregnancy, while that pubis becomes more flexible, as I mentioned, it is our center of gravity, it is our center of balance, so it also affects the female's gait. So typically a female who's late stage pregnant gets a very distinctive gait to the way she walks because of that loosening of the pubic symphysis. And notice I didn't say waddle. All right, questions on that? Excellent. So let's jump back and go back to all of that. So there you go. That is our cartilage. Notice they all have a matrix, they all have lacunas, they all have chondrocytes. And it is the condition of their matrix that is going to help us to tell them apart. And then we also know where to find them as well.
For the hyaline cartilage, is the perichondrium layer outside of the chondriocyte, like the layer, like the
Great
cell?
question. So let's cheat and do this. Oops. Sorry, hold on. Too many buttons. All right. So as we've mentioned, cartilage is avascular. So it's a solid matrix. It has its lacunas. It has its chondrocytes inside of the lacunas. And notice, unlike the osteocytes, it's much easier to see the nucleus of the chondrocytes inside their lacunas. But as I mentioned, it is avascular. So that means we need to some other way to be able to bring blood vessels close to it so it can get the oxygen and nutrients it needs. Or maybe we need a way to attach it to the material around it and stuff like that. So as it turns out, hyaline cartilage, and really all cartilages, have this other connective tissue that we haven't talked about yet on its outer surface. And this other connective tissue on the outer surface is a dense, irregular connective tissue. We'll talk about that in just a minute here, actually. And this dense, irregular connective tissue serves several functions. It provides some protection for the cartilage. Like I said, it houses the blood vessels that support the cartilage. Because while the cartilage is avascular, it's not dead, it's living tissue. So we need to be able to support it. We need to provide that protection. It houses the immature cells that allow the cartilage to grow and repair, even if slowly. And so we have this special tissue that wraps around cartilage. It's a dense, irregular connective tissue. Now, in fairness, we have not talked about dense, irregular connective tissues yet. But do you think wrapped around cartilage is the only place in the body we're going to find dense, irregular connective tissues?
No.
No, of course not. So as we've learned before, we can give things names based on their locations. So since I wrote it before, I'll abbreviate it here. A dense, irregular connective tissue that wraps around a cartilage because it is around a cartilage. Guess what we call it based on its location?
A perichondrium?
Yeah. What does peri mean?
Surrounding or around?
Yeah. Yeah, exactly. Surrounding, around the perimeter of, right? Perimeter, surrounding. Perichondrium. So it is around the cartilage. So the pictures that we had were focused on the tissue and not on the surrounding. Although if we have a chance to do histology, like I said, we'll talk about this, but on the exam, I could have a tissue like this. Again, it would be a histology slide. It wouldn't be my drawing, but I could have an arrow pointing here and I could basically ask two questions. Identify the tissue type, and you would say dense irregular connective tissue. Or I could say identify the tissue based on its location. And then you would say perichondrium.
So for the hyaline cartilage tissue type, that would be pointed to like, I guess inside, like near the matrix
No,
and the
outside.
hypochondria
It's the
site.
here, hold on. Like I said, I wasn't going to do this till later, but
Sorry.
no, it's okay.
Okay.
It's not a problem. Like I said, remember, I wanted these lists to be all inclusive. So that's why even if it has material we haven't talked about yet, we are going to get to it. So now is as good as time as any while it's fresh in our mind. Notice here, this right here is some hyaline cartilage. Anybody know what this out here might be?
Looks like,
Like a muscle tissue.
Yeah, exactly. It's actually skeletal muscle. So here we have our hyaline cartilage. And in this case, it is connected to some muscle. So notice here in this space in between is a tissue that surrounds the cartilage and attaches it to the tissue that it's next to. So on the exam, if I had this picture and I had this arrow, and I had this arrow, and I ask you to identify the tissue type at the tip of the pointer, what would your answer be?
Dense irregular connective tissue.
And if I asked you to identify the tissue based on its location, what would you say?
The perichondrium.
There you go. Like the name indicates, it's around the outside. It's the perichondrium. It's around the cartilage.
And then you would point inside, like for the hyaline cartilage, it would be right.
Yeah, so
kind of.
identify the tissue.
Okay.
No, so identify the tissue.
Oh, hyaline cartilage.
Identify the space.
The lacuna.
Identify the cell.
The chondria site.
That's exactly what the lab exam is going to be.
Okay.
It's going to be the vast majority of this lab exam is going to be histology pictures with arrows like that, asking these types of questions.
Okay.
Yep.
Thank you.
Yep. No, great question. All righty. Awesome. So those are our three types of cartilage. Any other questions on that? All right, let's go from our cartilages to our dense connective tissues, or remember, you can also call them fibrous connective tissues. And as the name indicates, these are very few cells and they have lots of fibers. And since they have lots of fibers, there's basically two ways we're going to identify them. Here, I'll cheat and I'll do it here. The first is going to be the actual types of tissues. Pardon me, types of fibers. And the second is going to be the orientation of the fibers. Take, for instance, our first one, the dense regular connective tissues. As you look at the illustration, and I'll cheat, notice for the most part, all the fibers are parallel. So we have a massive number of collagen fibers that are parallel. So it's a dense regular connective tissue. Now, what did we say the advantage of collagen fibers were?
Have tensile strength.
Excellent. So if we are pulling on this fiber, like that rope, pulling from one side and pulling from the other side, are we going to be able to tear this dense regular connective tissue?
No.
No. And so not surprisingly, this is what we use to form ligaments and tendons. Now, that does bring me to one important question. What the heck is the difference between a ligament and a tendon?
Doesn't one attach to bone while the other attaches to muscle?
Yeah, exactly. One attaches muscle to bone. Which one attaches muscle to bone?
Tendon.
Exactly. Whereas ligaments attach bone to bone. Under your kneecap, you have a tendon called the patella tendon that basically attaches your quadricep muscles, your big, huge anterior thigh muscles, to your tibia, your lower leg. Your muscles are so strong and your tendon has so much tensile strength that if you produced enough force in your quadricep muscles, you would actually break your tibia before you would tear your patellar ligament. You could actually produce so much force in your leg that you could actually break your bone, and the bone would break before that ligament would tear, or the tendon would tear. Right? We have these Achilles tendon, the Achilles tendon. We have the anterior cruciate ones in our knees. Incredibly strong, incredibly powerful. Well, if they're so strong and powerful, then how is it that athletes tear them?
Maybe overuse.
Some of it could be overuse, but notice also, as we mentioned, this tensile strength is only in one direction. When people tear their Achilles tendon, or when people tear their cruciate ligaments in their knees, it's typically because the force is coming from a different direction. They're shifting or trying to pivot in their movement, or that linebacker's hitting them from the side. Notice this only provides protection in one direction. So while it is incredibly strong, incredibly powerful, it only prevents stress in one direction. So we can use it for ligaments and tendons. Over the top of our head, over our belly, we have big, broad, flat tendons, which we give a fancy name to. An aponeurosis is a big, broad, flat tendon. But they only provide protection in one direction. That's great because my muscle only pulls in my leg in one direction. But what about my skin? Do I only stress my skin in one direction? Or do I stress my skin in all sorts of directions?
All directions.
All directions. So notice for something like my skin, I would need a dense, irregular connective tissue. Notice again, when we look at the illustration, when we look at the light microscopy, we have collagen fibers going in all sorts of directions. And so because these collagen fibers, again, it's still collagen fibers, but in this case, they're in all directions. And this gives us multi-axial. That multi-axial strength. It resists sharing forces in multiple directions.
So we wouldn't be able to see the cell, the fibrocyte, right?
No, great question. So absolutely, you can if we look closely, and that is a big key that we're looking for here. What color is going to show up really well? Let's see if this yellow shows up well.
Sorry, I can't tell from these pictures.
No, I understand. They're small. Notice, let's look at the light microscopy, the actual pictures. Notice one of the keys that's going to help you to easily identify a dense irregular connective tissue is the fibroblasts, the fibrocytes are big, large cells that run parallel. To the fibers. This dense irregular connective tissue in our skin also has fibroblasts and fibrocytes, but notice they're the little dark spots. Are they big and huge and large and elongated?
No.
No. So here, the nuclei are very small spots. And notice one less thing, fun with vocabulary. Even in our dense regular connective tissue, this cell right here that I just highlighted, is that one the fibroblast that is making the fiber, or is that one the fibrocyte that is maintaining the fiber?
Fibrocyte?
Anyone want to guess fibroblast?
Can you just
Fibroblast?
repeat the question?
So this cell that I just highlighted, this one right here, is it the mature cell that is making, that is maintaining the fiber, or is it an immature cell that's making a fiber? Notice you guys have confusion because you don't know. Well, guess what? I don't know either. There's no way to tell. So actually with our fibrocytes and fibroblasts, they pretty much use the terms interchangeably. Because when we look at them in the tissue, there's no way to know which is which. So you'll see that they use the terms completely interchangeably for these two, because there's no way to tell them apart. Osteoblasts, osteocytes, chondrocytes, those mature cells are easy to tell because they're inside the lacuna. But with the fibroblasts and fibrocytes, there's no way to tell them apart. So you can use the terms interchangeably. Now, remember I said we identify our dense connective tissues by the types of fibers and their orientations. And notice so far, both of these have had collagen fibers. So not surprisingly, there is a third type of dense or fibrous connective tissue called an elastic connective tissue. Notice this is not the same as the elastic cartilage. But elastic connective tissue and elastic cartilage do have something in common. And guess what the two things they have in common are?
They contain elastin.
Yeah, both of them contain elastic fibers. And notice here with our elastic connective tissue, they are elastic fibers and they are, again, mostly parallel. Now, remember, I mentioned that the nice thing about connective tissues is that they all look very different. You are never going to confuse hyaline cartilage and bone. You are never going to confuse blood and a dense irregular connective tissue. They look dramatically different. The only exception are these two fibers right here. Dense regular connective tissues and elastic connective tissues look similar. Both have parallel fibers. So we need a way to tell them apart. Notice with our dense regular connective tissues, typically the fibers are thinner and more precise, more sharp in their edge. Whereas here in our elastic fibers, the fibers are typically larger and more coarse. And what's the other big difference you notice between the dense, regular, and the elastic connective tissues?
The fibrocyte and like the nuclei.
Exactly. Notice our elastic tissue have the small pinpoint nuclei, whereas as we talked about up here, the dense regular has large parallel nuclei. And there is one third characteristic. Not always, but sometimes with certain stages, with certain stains, our elastic fibers will be yellow in color. It is not always the case, but when you see it, it is a dead giveaway you are looking at an elastic connective tissue. What type of tissue do you think this one is?
So dense fibers.
Dense
connected
regular.
Dense regular,
irregular.
absolutely. Notice mostly parallel fibers, elongated nuclei. Notice this stain, you don't see the fibers as well, but what do you see really, really well with this one?
The nuclei.
The nuclei, big, elongated, parallel nuclei. Notice here, we have fibers that are mostly parallel. But notice the edges are much more coarse. The fibers are much thicker. And when you look at the nuclei, are they big and are they parallel?
No.
No. So what type of tissue is this?
Elastic.
Elastic. And what do you notice on this one?
The yellow stain.
The yellow fibers. You see yellow fibers, that is a dead giveaway. You don't have to look at anything else. That is a dead giveaway. You were looking at an elastic connective tissue. Again, notice not every stain shows it this way, but when you see it, dead giveaway, which is good. On the exam, you want to have those dead giveaways that are going to help you to quickly identify something as you're going through them. All right. Questions on that?
What would a dense irregular connective tissue look like? Just that one. Okay.
So here, notice collagen fibers at all sorts of different directions. Notice again the small pinpoint nuclei. Notice this one doesn't show, although it does show, actually this one shows the nuclei pretty well. But notice again, all these collagen fibers at all these different orientations. That is this stain specifically stains the collagen fibers and shows them at all these different orientations. So you can clearly see this is a dense irregular. All right. Excellent. Questions on our dense, on our fibrous connective tissues? All right. From there, we go to our loose connective tissues. As the name indicates, there are fewer fibers. So there's more room for cells and more room for other stuff. And in particular, you see this in this tissue here. This tissue here that you're seeing is an areolar connective tissue. Now, it does not always have this appearance. But why does it keep doing that? Does not always look this way. But when you see this kind of cobwebby appearance, this is a dead giveaway that you're looking at an areolar connective tissue. This is by far the most common type of connective tissue. And remember, as we said, connective tissues are the most common types of tissues. So if you were randomly to reach into a body and pull out a tissue, what is it most likely going to be?
Areolar tissue.
Yeah, and again, I don't care how
It's
you say it as
connective.
long as you spell it correctly on the So again, are you likely to reach in the body and randomly pull out a tissue? No.
No.
But on an exam, if you don't know what the answer is, what should you probably guess?
This
Areolar.
one.
Areolar connective tissue, because it's the most common one. Absolutely. Notice, obviously, it has cells. We see all the nuclei here of the cells. That yellow is not showing up well. All the cells that we see in here. And notice also, we see a lot of fibers. Notice some of these are big and pink. These are like the collagen fibers we saw in our cartilage. Notice some of them are thinner, more fine and darker. These are the elastic fibers. So notice we have both collagen and elastic fibers in here. But there's also space for a lot of stuff. Some of that stuff are cells, but some of them can be, adipose, blood vessels, nerves, all sorts of things can be found in here. This is our body's bubble wrap. It surrounds and supports and holds our tissues in place, helps to hold our organs in place. And with all this extra space, this is a location where we could accumulate excess fluid and excess salts. And what do we call that when an area accumulates excess fluid and salts? Like after class today, falling asleep because I was up late grading your papers, so I need a good way to wake myself up. So I put my hand on the door frame and slammed the car door on my hand. That's a nice, refreshing wake up. And by the time I get to the store, what's happened to my hand as a result of that?
Swelling.
Swelling.
Yeah.
Yeah, swelling.
Edema. Absolutely. Right. This excess space in the areolar connective tissue is where our edema, where that swelling occurs. And thinking of it this way, is there any part of your body where if you damage it, it doesn't swell?
No.
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 So is there pretty much anywhere in the body where there isn't some areolar connective tissue?
No. What about your eyes?
Well, so maybe not the eye itself, but surrounding the eyes, is there areolar connective tissue? Yeah.
Yeah.
Yeah, maybe not inside, right? There isn't any inside the bone either, but again,
Right.
it's the most common tissue in our body. So absolutely, it's all over the place. All right, now notice one last thing about our areolar connective tissue. Notice there are all these different cells that we can see inside of it, and a couple of these cells are some adipocytes, some fat cells. What can happen is our areolar connective tissue can fill up with adipocytes. All right, someone's making a lot of background noise, so I'm going to mute everybody again. But again, feel free to unmute if necessary. We take that areolar connective tissue and we fill it with fat cells. And when we do that, we get our adipose tissue. Again, as I mentioned, all of our tissues look very distinctly different from each other. And adipose is very easy to recognize and identify. Notice a couple things here. We have all these very clear looking cells. Remember, as we've talked about, color doesn't matter on histology except when it does. In this case, the reason these cells are so clear is they're filled with basically an oil droplet, what is known as a fat vacuole. And this fat vacuole has a massive amount of oil inside of it. And oil, most stains that we use on our tissues don't attach to oils. So notice here, the cells themselves are completely clear. It kind of looks like chicken wire where we can only see the outside. And notice one other really important thing. Notice that when you have this big, huge, thick, fat vacuole in here, it pushes the nucleus way to the side of the cell. So notice the nuclei we can see have been shoved way to the side. It actually gives these cells a shape that they call a engagement ring shape, where you've got this cell where basically the fat vacuole has filled it up so much that it pushes the nucleus way to the side, making it kind of look like an engagement ring. Now, we know something special about these adipocytes. If you are eating 17 cheeseburgers for breakfast, lunch, and dinner, chances are you are taking in more calories than your body is using. And so what do these adipocytes do in that case?
They expand.
Exactly. They add more oil inside and they are going to expand as a result of that. If instead of 17 cheeseburgers for breakfast, lunch, and dinner, you instead eat 7 pieces of popcorn for breakfast, 7 more for lunch, and 7 pieces of popcorn for a very reasonable dinner, what's going to happen to your fat intake, your caloric intake as opposed to your caloric output?
They would shrink because they would use
shrink,
the fat.
exactly. We would have to use that fuel and the cells would shrink inside. So notice we primarily add fat and lose fat, not by adding more cells or getting or taking away cells, but by changing the size of the cells. Now, you may not have thought of it in those terms, but I know you're aware of it, because maybe you really like eating 17 cheeseburgers for breakfast, lunch, and dinner, and you don't want to change to popcorn. And so instead, right, because as we know, it's only on the outside that matters. So you go to your plastic surgeon and they have this nice big vacuum that they can stick underneath your skin and suck all those adipocytes out of your body. And what do they call that process?
Liposuction.
Liposuction. And once they get rid of all those adipocytes, you can go back to eating 17 cheeseburgers for breakfast, lunch, and dinner every day, because one of the amazing things about your adipocytes
is you don't make any more.
They do not divide. Adipocytes do not divide. So if I have my liposuction, can I go back to eating 17 cheeseburgers for breakfast, lunch, and dinner and not worry about getting fat?
Yeah, but your arteries will suffer.
Okay, true, my arteries will suffer, but will you really not get fat?
No.
No.
Why not? What's going to happen?
Wouldn't the remaining cells just get bigger?
True. Maybe they weren't as good at vacuuming, and so some cells left. But let's say, so again, this is a completely theoretical situation. Let's say that they actually were able to remove all of the adipocytes from your subcutaneous layer. Could you then eat 17 cheeseburgers for breakfast, lunch, and dinner and not get fat?
No.
No, it has to go somewhere.
True. And so you're right. You would get fat, but how would that happen? You would indeed get fat again as a result of that. But if our adipocytes can't divide and you have no adipocytes in your body, how is that possible? Where would those adipocytes come from?
Maybe like your organs?
True. So maybe some of the adipocytes could shift their locations. That's not a bad guess. We know all cells come from previous cells. We know adipocytes can't divide, but didn't we learn something about connective tissues? isn't there a pluripotent stem cell that can form all of the cells of connective tissues?
Yes.
What was that one again? There we go. Absolutely. Even if you were able to theoretically suck all of the adipocytes out of your body, we still have mesenchymal stem cells. So as you continue to eat those 17 cheeseburgers for breakfast, lunch, and dinner, that would signal your mesenchymal cells that we need more adipocytes, and that pluripotent stem cell would divide to make more adipocytes to replace the ones that were lost. Yeah. So even liposuction is not the answer. There's no good way to eat 17 cheeseburgers for breakfast, lunch, and dinner. Trust me, I've tried. All right. Questions on that? All right, excellent. All right. Oh, what is the function of our adipocytes?
Is it for energy?
Yeah,
Yeah,
absolutely.
Store and storage.
Absolutely, it is storage for energy. Anything else?
A protection layer.
Yeah, it provides protection, both mechanical protection and thermal protection, helps to insulate the body that way. Again, as we mentioned, we'll find adipocytes all over the place. One of the important places you find adipose is around your kidneys, providing structure and support for the kidneys. One of the things when people go on those dramatic weight losses, like they do the stomach stapling or banding, or they go on the show like The Biggest Loser, one of the things that the doctors are watching very, very closely is their urine output, because your kidney is surrounded by a big fat pocket that basically holds it in place. All right, the kidney is obviously where we filter our blood, end up making urine, and then that urine leaves the kidney via a vessel called the ureter to go to the kidney, to the bladder, so that it can be expulsed from the body. With regular, normal weight loss, healthy weight loss, it is slow enough where your adipose is actually very dynamic. Fats are constantly being bundled up and shipped to different parts of your body as needed and to help to be maintained. But when you do rapid and dramatic weight loss, you can lose adipose in more places than just subcutaneously. And if you're losing weight too rapidly, that adipose that's holding your kidney in place can start to go away, and the kidney can actually sag in your abdominal pelvic cavity. It's a condition they call a ptosis. Now, the reason this is a concern is if it sags, it can actually cause the ureter to kink. And what happens if you kink a hose?
You can't get the water out.
Yeah, not only does the water not go out, but the water starts to back up the hose, and you get an increase in pressure in the kidney, and that can cause major and permanent damage to your kidneys. So they monitor their urine content, the urine output of these contestants on shows like The Biggest Loser, to make sure that ptosis hasn't occurred, to make sure that they maintain the health of their kidneys. So again, from a social standpoint, we just think of fat as, the subcutaneous stuff. It is very important and very vital for our function. Lastly, you guys mentioned earlier you wanted to see what a reticular fiber looks like. Again, notice here we see these short, elaborately branched reticular fibers made of collagen. I love this. It always reminds me, for those of you who ever been back east or like Washington, D.C. or something like that, cherry blossom trees. The cherry blossom trees have these elaborately branched, very beautiful branching, and these pretty little pink blossoms on it. And this reticular tissue always reminds me of that. Notice the advantage of this, unlike our dense regular, where we have all these fibers that are all straight, These are short and elaborately branched. And so notice there's a lot of space in here. And all these dots. Wow, that guy's really annoying. Whoever that is that's talking is really, really annoying. Oh yeah, that's me. These little dots are all like, for instance, white blood cells housed in these areas, providing protection. So this provides very soft, very porous tissue. So like I said, we find this in our lymph nodes, we find this in our liver, we find this in bone marrow, and we find this in our spleen. These are all areas where we have this very distinct reticular loose connective tissue. All righty. Lastly, blood. As I mentioned, when we get to 431, in fact, if you take me for 431, the very first system we'll do will be the cardiovascular system. And we will talk about blood in great detail there. For now, though, we still need to know it is a connective tissue. Again, it's sometimes referred to as vascular tissue because it's inside the blood vessels. And it is indeed a connective tissue. Remember, to be a connective tissue, you have to have cells and you have to have a matrix. Let's start easy. Does blood have cells?
Yes.
Yeah, like what?
Red blood cells. White blood cells?
Anything else?
Hemoglobin?
Hemoglobin's in the red cells. How about platelets?
Yeah.
So those are
That's
three
one, yeah.
things, absolutely. So we have cells here. However, look at our picture up here. Notice, here's a red blood cell. Does that red blood cell really look like a typical cell that we learned about in the last section?
No.
No, there's no nucleus. There's no organelles. In fact, as someone mentioned, they're basically just big bags of hemoglobin. At one point in their life, they had nuclei. They were able to make proteins. They were able to divide. They're able to do anything like that. But as mature red blood cells, they're not really true cells. Now, by comparison, when we look at a white blood cell, do those have nuclei? Do those have organelles? Can they divide? Can they make proteins? Can they do all of those things?
Yeah.
Yeah, so they are indeed true cells. But what about a platelet? Do we see even a platelet on this? Oh, we see a little piece of dirt or something that they've got on the slide. Isn't that what that is? A piece of chewing gum on the sidewalk? No, that's actually a platelet. A platelet is actually a cell piece. There's a big, huge cell called a megakaryocyte. You don't need to know that name. But basically, the little pieces of it get ripped off. And when they get ripped off, they basically become the platelets. So notice, a platelet's not a cell. It's really a cell piece. So notice, two of the three things that we're calling cells aren't really true cells. They used to be true cells. They came from a true cell, but they're not a true cell. Now, there are white blood cells that are indeed true cells, so that does meet our criteria, but it does make labeling a little awkward. So typically what happens is, rather than using the term cells, we instead use the term formed elements. Formed elements is just a fancy way of saying cells and things that used to be cells. So when we talk about the cells of blood, we instead use the term formed elements, but they're there and they match. And of course, as we also talked about, and I think someone, oh yeah, the two mentioned it, we have a matrix as well, and the matrix is indeed the plasma. Remember, matrix is ground substance, which is basically water and stuff. And then we also have fibers, and we learned how there are water-soluble fibers. So yeah, absolutely, blood meets our criteria. It is a connective tissue. It has matrix. It has cells. And there's one more thing we have to do as well. while we need to know this is a connective tissue, it has formed elements, it has matrix, this is an anatomy and physiology class. Are we going to get away with terms like red blood cells, white blood cells, and platelets in an anatomy and physiology class?
No.
No.
Yeah. So on the exam, if I have an arrow and I have it pointing to this formed element on the exam, and I ask you to identify the formed element, what would your correct answer to that be? What's the
Red blood
appropriate
cell.
anatomical term instead of red blood cell?
Oh, red blood cell.
Yeah, what's the appropriate anatomical term for red blood cell? Pretty sure if you have your connective tissue handout in front of you, you might be able to figure this out.
Erythrocyte.
There we go. Erythrocyte.
Wait, so what is a thrombocyte?
What do you think the thrombocyte is?
The cell.
Nope, platelets.
Right.
And what is the general term for a white blood cell?
Leukocyte.
Leukocytes. Excellent. Now, be careful, Sarah. There are actually 5 different types of leukocytes. You don't need to know those for the exam. Lymphocytes is an example of one of those. But again, the general term for a white blood cell is leukocytes. So erythrocytes, leukocytes, and thrombocytes are the appropriate anatomical terms you will want to use. Do not use red blood cells, white blood cells, and platelets on the exam. You will get that wrong. So use the appropriate anatomical terms. And then obviously this stuff here that looks clear isn't really clear. That's the plasma, that's the matrix. All right. Questions on that? Excellent.
I have a question.
Yes, go ahead.
So this is kind of farther back, for the areolar connective tissue, I was wondering what a mast cell is and what it would look like. And then for the histamine granules, we obviously probably can't see it. So will we just assume that it's there?
You can see it, which is how
Oh, you
we're going to
can.
be able to tell what that is.
Okay.
Let's wait. Oh, actually, I'll tell you what, we'll go ahead and do that now. Because I don't know if we'll get a full formal histology review at the end of this. So might as well answer this question now. Let's see. Okay, perfect. Identify the tissue type.
Areolar.
Yeah. How do you know?
Cobwebby looking.
Exactly. So there's 3 ways you should have come up with that answer. 1, you can see that it has that cobwebby appearance, which we said is a dead giveaway that it's an areolar connective tissue. 2, Ava specifically asked about the areolar connective tissue, so that makes sense that would be the one we were talking about. And 3, remember, areolar is the most common, so if you don't know what else to guess areolar. Excellent. So this is an areolar connective tissue. As I think Sarah pointed out, it has that cobwebby appearance. And notice there are a ton of cells all over the place. Normally, when we see the cells, we can just see the nuclei and not much else. Now, as we look at the nuclei of these, especially at such a low magnification, is there a way to tell what these particular cells are?
No.
Maybe, but do I expect you to be able to do it?
No.
No, absolutely not, right? So I don't expect you to be able to do that. However, notice when we look a little closer, again, we see lots of cells. None of these are showing up with green. Here we go. Let's see if that works. There we go. There and there and there and there. And again, normally we just see the nucleus. And again, is it possible for someone to tell what these things are? Yes. Do I expect you to be able to do it? No. However, if you noticed 3 of these things are not like the others. This cell, and this cell. Notice, not only do we see the nucleus of this cell, let's actually make that darker, but notice also we see the entire cytoplasm of this cell. And this cytoplasm is very dark, very distinct, and very granular. The reason we're able to see these cells is because they are chock filled with very dense granules containing a very specific chemical. And what chemical do you think these cells have inside of them?
Histamine.
Histamine. And any idea what histamine does?
I've heard of it, but I don't know.
Yeah, I want to say it helps with it releases when you have like an allergic reaction.
Or like inflammation?
Absolutely, you're both absolutely correct. Histamine is one of the chemicals, one of the main chemicals that controls inflammation. So like we talked about, if I slam my hand in the car door, the damaged tissues, the mast cells in this area release histamine. And that histamine causes blood vessels to dilate. That brings more blood to the area. So the area gets red, the area gets warm because blood brings that heat with it. And it makes the blood vessels leaky. So more fluid comes out, I get the swelling. And when the tissue swells, it pushes on the nerves and I get pain. So notice redness, heat, pain, and swelling are the 4 cardinal characteristics of inflammation. Now, as someone pointed out, slamming my hand in the car door damages the tissue, and it is appropriate for that histamine to be released. But if someone's mowing the lawn, right, that cut grass can incorrectly stimulate some of my cells, causing the cells in my nose to increase blood flow, become more leaky, and suddenly my nose becomes congested and my nose starts running. And so what can I take to combat that?
Antihistamine.
An antihistamine, absolutely. So
And
that
these are all positive feedback, right?
no, it's not a positive feedback. It is a process to help to bring it's a repair process. So again, homeostasis is about maintaining balance. If the tissues become damaged or injured, then we need to respond appropriately to repair that. So that's more part of the repair process for doing those types of things. And then an allergic reaction is basically when you get that repair process inappropriately. All right. So these cells chock filled with histamine make them stand out, make them look different from all the other cells we're responsible for or could tell, and make them easy to identify. So what are these easy to identify cells inside of our areolar connective tissue? Ava, what did you ask me about?
Mast cells.
Mast cells. So guess what these 3 cells are?
Mast cells.
Mast cells. And what is the substance? What is the chemical they contain inside of them?
Histamine.
There you go. Did that answer your question?
Yes, thank you.
Excellent. All right. Any other questions? It's a great question. Any others? I like that you're looking at your list and figuring out what it is that you need to be responsible for. That's excellent. All right. So now we understand epithelial tissues. Now we understand connective tissues. So what we need to do now is put them together to make a membrane. And that's exactly what a membrane is. A membrane is a superficial epithelial tissue on top of a deep connective tissue. And that's why, like I said, we have to talk about epithelial tissues and connective tissues first, because once we know both of them, we can put them together to make membranes. And that's what we're going to do next. Let's go ahead and take our second break first, though. So that it is 10:40, so we will come back at 10:55 and restart, and I will start the recording at that time. If you haven't caffeinated yet, I will strongly encourage you to caffeinate now. Again, one of the things that we've talked about in this class is learning anatomy and physiology is like learning another language. And when we're talking about these membranes, there is a massive amount of vocabulary and a massive amount of conditional vocabulary. Remember, there are many things we name based on their location. All right. There is truly nothing, well, actually, it's using an even better example. There is truly nothing special about my AirPods, right? There are a gajillion of them in the world. However, these happen to be in my hand. They're sitting on my desk. So based on their location, they're Daniel Slutsky's AirPods, right? And we see the same thing with tissues. We talked about a perfect example of that earlier. A dense irregular connective tissue that is around cartilage, we call a perichondrium, right? A serous membrane that is around the heart, we called a visceral pericardium. This idea of giving things names based on their location is not new, but it's something we're going to see a lot in these membranes. So let's go ahead and take our break, 15-minute break, come back, and we will finish off our discussion of tissues by finishing off our discussion of membranes. All right, see you guys in 15 minutes. Let us go ahead and get started. So, as I mentioned, we identify a membrane as a superficial epithelial tissue on top of a deep connective tissue. And all of the surfaces of our body are basically covered by one of four main types of membranes. Now, some of these we've already talked about a little bit. We've definitely talked about serous membranes. We talked about them a lot and where they're located. I think we were talking about mucous membranes when we were talking about the digestive system. What's the other name for our skin? What kind of membrane is our skin?
Cutaneous.
Yeah, cutaneous membrane. And inside of our free moving joints, we have a synovial membrane. So there are four main types of membranes: a mucous membrane, a serous membrane, a cutaneous membrane, and a synovial membrane. So we're going to talk about and identify all of these. So let's move to the whiteboard real quickly. We're going to talk about our membranes. With, again, a superficial epithelial tissue and a deep connective tissue. We're going to identify their locations, their functions, and their anatomy. So, as we talked about, there are mucous membranes, or again, fun with vocabulary. Mucus is the adjective that describes the noun membrane, or we could use the noun mucosa. So mucosa is the noun for that. We talked about how there are serous membranes. And I think we've done that already. Serous membrane. Serous is the adjective describing the noun membrane, and serosa. There is our cutaneous. membrane. And there is our synovial membrane. So those are our 4 membrane types, and let's talk about each one. And we will talk about mucous membranes first. So as I mentioned, when we're doing this, we are going to want to identify first their location. How would you guys describe the location of a mucous membrane?
In the single columnar.
Well, again, I don't want to worry about the tissue type yet. We'll get to the tissue type later. But give me an example of some place where you find a mucous membrane. Can you give me any example of some place where you find a mucous membrane?
Nasal cavity,
Nasal cavity, excellent. Where else?
lungs.
Esophagus.
Lungs, esophagus, absolutely. Notice what these things have in common. All of these mucous membranes line cavities that are open to the outside world. So, what organ systems are open to the outside world?
The respiratory and the reproductive.
Respiratory, reproductive,
Digestive.
digestive.
Integumentary.
Oh, true, but although remember, integumentary is cutaneous membrane, so that's not a cavity per se. So we're missing one though.
Urinary.
Urinary, there you go. Excellent. So notice those 4 organ systems: respiratory, digestive, reproductive, and urinary. All of those are open to the outside world, and so they're all lined by mucous membranes. And of course, what is the function of a mucous membrane? All right, you guys are definitely overthinking.
Protection.
How?
Mucus.
Yeah, it makes mucus. Absolutely, it makes that mucus. And you're right, mucus provides protection, lubrication, right? Capturing dust and debris, all of that kind of important stuff.
So when
Excellent.
you're asking us location, you want us to describe like the cavities or the systems that they're found in?
Well, it depends on the question, obviously. If I were to ask you one location where you would find a mucous membrane, then yes, you could say the oral cavity or the nasal cavity or the esophagus or the lungs or the vaginal canal or the urethra or the anus or, small intestine, large intestine. Any of those organs that are open to the outside world would be a location. This location here, I was thinking just in terms of general terms, where is a mucous membrane found? All right, we're describing mucous membranes in general. They're line cavities that are open to the outside world, and they make mucus. Right? Now, as someone pointed out, if we look at the pretty picture from your textbook, we see an example of one of those, like the one we found in our stomach or our small intestine, where the epithelial tissue was a simple columnar epithelial tissue. So notice again, if we go back to our whiteboard here, when we talk about the epithelial tissue, right? You could remember it was epithelial, simple columnar, because we talked about it when we were talking about the stomach and the small intestine. You could have cheated when you looked at the picture for this lecture slide and be able to see that. However, we just mentioned 4 organ systems that are open to the outside world. Are all 4 of them lined just with a simple columnar epithelial tissue?
No.
No. What else then can help to form a mucous membrane?
The ciliated pseudostratified columnar.
Excellent. Ciliated, pseudostratified, columnar, epithelial tissue, like in the nasal cavity or a trachea. What else?
Transitional.
Transitional in our bladder, in our ureters, in the proximal part of our urethra. What else?
Non-keratinized stratified. Or yeah, stratified squamous. Epithelium.
Excellent. What about in the lungs where gas exchange takes place?
Oh, isn't that dense or regular?
Well, again, let's not talk about the
Connective tissue?
connective tissues. What epithelial tissue did we say you had in the lungs for the rapid diffusion of oxygen in and out?
Simple squamous.
Yeah.
Oh, I'm okay. Yeah, I think that I'm thinking.
Yeah. Notice pretty much almost any epithelial tissue can be part of a mucous membrane. Right? The key is that it depends on the function. As we just finished talking, in our lungs, we want that simple squamous for rapid gas exchange. But in our nose, we want that ciliated pseudostratified columnar so that we can move the mucus. In our oral cavity, we want that non-keratinized stratified squamous to protect us from that hot coffee that we're having for breakfast, right? And so on and so forth. So it really is going to depend, it's going to be almost any epithelial tissue, and it really depends on function, right, as to which epithelial tissue it's going to be. However, no matter what is on top of it, there is one and only one connective tissue that will form the base of a mucous membrane. And what do you think that one and only one connective tissue that will form the base be?
The dense, irregular.
Not a bad guess, but what's even more ubiquitous, found almost anywhere?
Oh, the collagen. Oh, wait.
Areolar?
There we go.
Ah,
It's going to be the areolar connective tissue.
I was thinking how collagen proteins are.
Collagen is the most common protein, but here we're talking about
Yeah.
the most common connective tissue.
Connective tissue.
Now, as I have mentioned, areolar connective tissue is found all over the place in the body. So this is just one location we'll find it. However, based on that, we want to give it a name based on its location. An areolar connective tissue that is part of a mucous membrane, based on its location, we call it a lamina propria. Oops. Notice if we go back to our lecture slide, as we talked about, it can be all sorts of different possible epithelial tissues, but only one connective tissue, no matter what is sitting on top. We know it's going to be a areolar connective tissue underneath. And as we mentioned, based on its location, we are going to call it a lamina propria. Again, I'll say this again, because despite all my warnings on the exam, people lost points on the lab exam for two main reasons, right? Again, obviously, some people got some things wrong, but a large number of you forgot to use left and right, despite all my warnings, and so lost a fair number of points for that. And some people didn't read the question carefully, and this is a perfect example. This picture could be on your exam. It's not, because it'll be a real tissue. But let's say for argument's sake it was. I could have an arrow pointing here, and notice there are two questions I could ask. I could ask you to identify the tissue type, and what would your answer to that be?
Areolar or connective tissue?
Yeah, areolar connective tissue. Or I could ask you to identify the tissue based on its location.
Lamina. Oh, sorry.
You know, go
Lamina
ahead. Lamina propria, absolutely. All right, and that's exactly how I will ask those two questions. I'll either ask you for the tissue type or the tissue based on its location. All right. And while we're talking fun with vocabulary, you'll notice I've written mucus, M-U-C-U-S and M-U-C-O-U-S. Which one is correct? Yeah, Sarah's got it right, both. Fun with vocabulary. U-S is the noun, right? I have a bucket of mucus. That is a noun. However, if I take that bucket and I dump it on one of my kids, then my kid would be described as being mucusy, and that's the adjective. Now, remember in this class, you do get one letter. So I guess it doesn't matter whether you spell it US or OUS, you're still going to get it right on the exam, but it's probably good to eventually learn the difference between the two. So I will mention it here. All righty. Questions on our mucous membranes?
Could you point an arrow to where exactly the mucous membrane is?
No, I can't, because the whole picture is the mucous membrane. Remember,
So
a mucous
when-
membrane is made up of an epithelial tissue on top of a connective tissue. So you have to have both. This picture has both. So the entire thing in this picture is the mucous membrane.
So when we're labeling on our histology worksheets, how do you want us to draw it? You just want us to,
Well, so probably the way I would
Or
do it,
label
and again,
it.
there's multiple ways to do this, but one of the easiest ways probably to do it is so let's say I was drawing this tissue, my simple columnar epithelial tissue. I'd make sure I had my nuclei all ducks in a row, and then I have my relar connective tissue there. And probably what I would do is I draw a bracket, making sure that I was pointing at the epithelial tissue and the connective tissue, and then write that this was my mucous membrane, because it's the whole thing. Or just have an arrow pointing to the whole circle, because it's the whole thing, right? Somehow indicate to me that that whole thing in the field of view, the epithelial tissue and connective tissue together, are your mucous membrane. Because remember, you have to have both. For a sandwich, you have to have the bread and the stuff you put in the middle, right? It's all together as a sandwich. Well, an epithelial tissue and a connective tissue together is what is necessary to make a membrane. All righty. Let's go to one we know even better, because we've already talked about it. Serous membranes, cirrhosis. How would you identify in general the location of a serous membrane? We know specifically where they were found: around the lungs, around the heart, around the stomach and the liver and the spleen and all of that. Is there a way that we could define that, maybe similar to the way we define mucous membranes, for consistency's sake?
Like surrounds body cavities or
Yep, surrounds cavities that are what?
enclosed or
Yeah, that are closed to the outside world, right? If your serous membranes are exposed to the outside world, then you need to do something about that. See a doctor immediately. And as we also learned, what is the function of our serous membrane?
make serous fluid.
Excellent. We want to make serous fluid. Why? What did that serous fluid do again?
It's used for like protection, for like friction, or
Yeah, exactly. It provided protection, reduced friction. Now, let's think about this. Do we want to fill this cavity, that serous cavity, with as much serous fluid as we possibly can? Is that going to be good for the lungs or good for the heart to have as much fluid as possibly we can around them? No. So notice what we really want is to make a very thin layer of serous fluid to reduce that friction, to provide that protection. Remember, we also have serous fluid in our saliva, in serous fluid a couple other places. So this thin layer of serous fluid that lines our cavities, we could give it a name based on its location. And so that thin layer of serous fluid on the top of a serous membrane, we actually call a transudate. So notice when we go to our picture from the textbook, here we have our serous membrane, and notice we have this thin layer of serous fluid lining our serous membrane. That's called the transudate. Now, we've learned about our serous membranes and we've identified them all by their location. And if you remember correctly, there's 2, 4, 8 different serous membranes, right? Visceral peritoneum, parietal peritoneum, visceral pericardium, parietal pericardium, right pleural visceral, left pleural visceral, right parietal visceral, left parietal visceral. So 8 different ones we've identified by their locations. But anatomically, they're all the same. They're formed by an epithelial tissue sitting on top of a connective tissue. Now, since all of these cavities are closed to the outside world, do we need to worry too much about protection from harmful things from outside the body?
No.
No. So notice all we need is a barrier, the simplest barrier we can possibly make. So based on this picture, and based on the fact that we know we just need the simplest layer possible to make that barrier, what epithelial tissue are we going to use to make a serous membrane?
Simple squamous.
Yep. Now, as we know, is this the only place in the body where we'll find a simple squamous epithelial tissue?
No.
No. So a simple squamous epithelial tissue that is part of a serous membrane, based on its location, we call it a mesothelium. Oops, hang on. And it needs to be sitting on a connective tissue. What connective tissues are going to sit on?
Areolar.
There you go. Now, is this areolar connective tissue a lamina propria?
No.
No. Remember, lamina propria is only an areolar connective tissue that's part of a mucous membrane. This areolar connective tissue is not part of a mucous membrane, and so it actually doesn't have a name we need to worry about. We just need to know that it's an areolar connective tissue. And that's what we see from the pretty picture. So doesn't matter whether it is lining the thoracic cavity, directly on the stomach, right, forming the cavity around the heart. Every single serous membrane we've talked about is just made up of these 3 things. A simple squamous epithelial tissue called the mesothelium based on its location, an areolar connective tissue, and a thin layer of serous fluid called a transudate.
So the mesothelium is always the one closest to the areolar tissue.
Yes, because the fluid is on top of the epithelial tissue.
Okay.
Remember, transudate is just fluid. Right. Whereas the mesothelium is an epithelial tissue, which is superficial, and it sits on top of an areolar connective tissue.
So an example of like the line cavity could be, let's say like the parietal layer of the pleura, for example. If you
Sure.
ask for
Actually,
location.
let's do this. Here, of course, as you can clearly tell, is a human heart. Right, that's exactly what it looks like. As we know, the human heart is lined by a, let's make this pink, visceral pericardium. And we now know that that visceral pericardium has two parts. It has a thin layer of areolar connective tissue. And on top of that is a simple squamous epithelial tissue. We know this is a double membrane. So we know out here around the heart is the pericardial cavity. And our pericardial sac. And we also know these two things are connected to each other. So our areolar connective tissue actually comes off the wall and lines the parietal pericardium, and our simple squamous epithelial tissue comes off and lines the cavity as well. And lastly, we know that both of these have a thin layer of serous fluid lining their surfaces, which is what makes them slippery, so that it reduces the friction as the heart is constantly beating. Again, we've talked about a visceral pericardium and a parietal pericardium before, but now we actually know the histology that makes it. So if you think about it, if you were doing heart surgery and you needed to get to the heart, you would have to first pass through, oh, hold on. You would first have to pass through the areolar connective tissue, then a mesothelium, then a transudate, then the empty space, then another transudate, another simple squamous epithelial tissue, and another areolar connective tissue to then get inside the heart. Of course, there's heart muscle and other stuff as well, but those are those two layers sitting on top of each other. All right. Questions on serous membranes?
So as for location, how specific do you want us to be? Like,
Depends on the question. If I'm asking you, as I could easily do, to identify and describe the 4 types of membranes that are found in the body, then the general definition for a serous membrane is that they line cavities that are closed to the outside world. If instead I'm specifically talking about the heart and what membranes are associated with the heart, well, then yeah, then you would say that specifically that is the pericardium, right? Or something like that. Okay. All righty. I mean, heck, let's look at one other example. Instead of the heart, let's do the stomach. In 30 seconds, I'm going to teach you how to make a stomach. As you know, of course, the stomach is connected to the outside world. So lining the inner surface of the stomach is a mucous membrane. And of course, as you know, mucous membranes are made of epithelial tissues sitting on top of connective tissues. What would the epithelial tissue be lying in the inside of the stomach? I'm not gonna be able to do this in 30 seconds if you guys don't answer my questions. What epithelial tissue do you find inside the stomach?
A simple columnar.
There we go. Excellent. What connective tissue is going to form the base of that mucous membrane?
Areolar connective tissue.
Excellent. And because this is an areolar connective tissue that is part of a mucous membrane, do we give it a name based on its location?
Yes, mesothelium.
No.
No, we don't.
Oh, mucous membrane. Sorry.
What
A
areolar
lamina
connective tissue?
propria.
There we go.
Lamina propria.
Excellent. All right. So that is what's on the inside of our stomach here. We have a simple columnar epithelial tissue. Sitting on top of an areolar connective tissue. Then, not surprisingly, we have some smooth muscle. All right, that's what allows the stomach to churn and do all of that. And then we have the outside of our stomach. Notice our stomach is inside the abdominal pelvic cavity. So what type of membrane do we have on the outer surface of the stomach?
Serous membrane.
Excellent. We have a serous membrane. As we know, serous membranes are made of epithelial tissues sitting on top of connective tissues. What would the epithelial tissue be?
Simple squamous. Squamous.
Excellent. And what do we call a simple squamous epithelial tissue that's part of a serous membrane?
Mesothelium.
Excellent. And what type of connective tissue is it sitting on top of?
Areolar.
Excellent. So notice we have a little bit of areolar connective tissue. And on top of that, we have a single layer of flat cells. Notice we're going to have a little bit of mucus on the inside surface of the stomach, a little bit of serous fluid that transudate on the outer surface. Oops. Mucus, oops. On the inner surface. And out here we have our serous fluid, which based on location, we call it transudate. And there you go. We just made a stomach. That is the histological anatomy of a stomach. A serous membrane on the outside, a mucous membrane on the inside, and some smooth muscle in between. And we just made a stomach. So all these things go together. All right. Questions on that? Excellent. All righty. From here, things get a teeny bit trickier. Let's talk about synovial membranes. And this may be one you're not quite as familiar with. Any idea where you find a synovial membrane?
In your joints or?
Yeah, exactly. This location is that they line the cavities of free moving joints. So like here, my finger, these free moving joints, or my shoulder, or my elbow, any of those free moving joints, they're there. And what is their function? What do you think the function of a synovial membrane is? Well, let's see. Mucous membranes make mucus. Serous membranes make serous fluid. What do you think synovial membranes do?
Synovial
Synovial
fluid.
fluid.
Why?
Because sometimes anatomists aren't that bad.
True, but no, but why do we need synovial fluid? What does synovial fluid do?
It makes the joints move easier.
Yeah, it reduces friction of the joints. Now, in this case, do we want a thin layer of synovial fluid?
Thick.
Thicker.
We want to fill the joint, absolutely. We want to make synovial fluid and fill the joint. We want to fill that cavity to provide that protection, that cushion, that lubrication for movement. Absolutely, excellent. Now, again, it is going to be made up of an epithelial tissue sitting on top of a connective tissue, but let's cheat and take a look at the picture associated with this. Notice here we see a free moving joint, and then they give us this up close view. Notice when we look at the epithelial tissues, normally we have a dense layer of cells, right, where they're all kind of the same shape and it's all continuous. Whereas notice here, there's a cluster of cells here and a cluster there and a cluster there. This is not a true epithelial tissue. Instead, what we have is really a clusters of epithelial cells. So it's not a true layer. Instead, it is clusters of cells. And so we give a fancy name to this. We basically just say it is a scant layer. It's which is just a fancy way of saying it is an incomplete layer. Of epithelial cells. And even the connective tissue is a little funky. Not surprisingly, it is again going to be an areolar connective tissue because it's an areolar connective tissue for everything so far. But in this case, it has a more extensive matrix, which is a fancy way of saying it has more fibers in it. Which makes sense. If I have a freely moving joint, I need to provide a little bit more strength, a little bit more integrity. So having a few more fibers in it or something that is going to be helpful. So absolutely, when we're here in our synovial membrane, we have that more extensive matrix with more fibers, a kind of incomplete epithelial tissue on top of it. But it still has epithelial cells, it still has connective tissues. It's still lining a surface, so we still consider it a membrane. All right, questions on that?
All right, can you go back to the previous slide where you had writing everything?
The whiteboard?
Yes, please.
Yeah.
Okay, got it.
Yep. All right, any questions on synovial membranes?
These are found in like joints.
Yeah, free moving
Okay.
joints, absolutely. Excellent. And so then that leaves us with our last membrane, our cutaneous membrane. And of course, what is the location of our cutaneous membrane?
The skin.
Yeah, your skin is the cutaneous membrane. Your cutaneous membrane is your skin. Basically, those are interchangeable words. Skin and cutaneous membrane mean the same thing. And obviously there's more to it than this, but in general terms, what's the function of your cutaneous membrane?
Protection.
Yeah,
Structure.
providing that protection from the outside world. Obviously there's more into it, but we'll get into that when we get to the integumentary system. What we can talk about now is, of course, as you know, what epithelial tissue it helps to form our cutaneous membrane. Remember when you go to England and the Queen gives you her hand, what are you supposed to do?
Guess it. So keratinized, stratified.
Keratinized, stratified.
Yes.
Oops. Excellent. And that epithelial tissue is sitting on top of not one, but two connective tissues. Now, based on everything else you see here on the list, what do you think one of those epithelial tissues is?
Areolar tissue.
Yeah, because it's everywhere else as well. However, as we talked about, the deep layer of our skin has to protect us from all of that stress at all those different angles. So what was the tissue that would help us with that?
A dense irregular connective tissue.
Excellent. And that finishes off our tissues and takes us to our discussion of the skin, our cutaneous membrane. So let's go back to our summary page where we've put all this piece stuff together. Here are the 4 membranes. We identified their general locations, we identified their general functions, and we identified their anatomy, as well as identifying all terms that are based on locations. And as we talked about before, is this the kind of thing that makes a good essay question?
Yes.
Absolutely. But this is also information that lends itself well to the lab exam. Because let's take it one step further. Right? If you remember before, one of the things that I could do is have this slide, this arrow, and ask you to identify this tissue type. And what would your answer to that be?
Simple columnar.
Simple columnar epithelial tissue.
Epithelium.
Don't forget to say epithelial tissue.
Can you say epithelial tissue or epithelium?
Correct. Epithelium is the noun. Epithelial
Great.
tissue is the adjective that describes the noun. However, notice if I take the arrow away, I could ask you to identify the membrane. And what membrane is this?
Mucous membrane?
Mucus, right? There's a couple ways you should be able to easily and quickly figure that out. One of these, again, a simple dead giveaway, is the presence of this cell. What cell was this again?
Goblet cell.
Goblet
What
cell,
What do goblet cells make?
Mucus.
mucin.
Mucin, excellent, which then hydrates to become mucus. So clearly that makes this a mucous membrane if it's releasing mucus. So that's easy. But remember also, where are the locations that we find simple columnar epithelial tissues?
And then in the stomach.
Stomach, small intestine, large intestine, which are all part of what organ system?
Digestive.
Digestive system, which is open to the outside world. And so that tells us that this must be a mucous membrane. And notice, once we figure that out, we can figure a couple other things out as well. Identify the tissue type.
Areolar connective tissue.
How did you know?
Because it's always going to be underneath
Yeah.
the epithelial tissue for the mucous membrane.
Notice this doesn't look particularly cobwebby, but we know if this is a mucous membrane, this must be an areolar connective tissue because that's the only connective tissue that forms it. And identify this tissue based on its location.
The lamina propria.
Here you go. Notice. Identify the membrane type.
It's a mucous membrane.
How do you know?
Because
How about
the
cells?
cilia.
Yeah, cilia means this is a ciliated pseudostratified columnar, which is either found in the nasal cavity or the trachea, part of the respiratory system. So that means this must be a mucous membrane, which also tells me identify this tissue based on its location.
Areolar.
Identify the tissue based on its location,
Oh,
Lamina
lamina
propria.
propria.
lamina propria, and identify the tissue type.
Areolar.
There you go. Identify the membrane type.
Cutaneous membrane.
Not a bad guess, but what do you notice when you look up here?
Oh, the nuclear flat.
Nuclei. So what type of tissue is this?
It's a non-keratinized.
Non-keratinized. And if this is a non-keratinized stratified squamous epithelial tissue, then what type of membrane must this be?
Mucus.
Mucous membrane, absolutely. All right. Because what are the locations where you would find this tissue?
Cavities are open to the outside.
Yeah, but give me one specific one.
Nasal cavity.
Mm,
Oh, I mean,
not nasal. How about oral cavity?
oral cavity in it.
Anus, oral cavity, esophagus, vaginal canal, right? All of those are places where we need to have that protection, reduce the friction or protection from friction, I should say. So, but you're right. These are all open to the outside world, which means this is a mucous membrane, which also tells us identify the tissue type.
Areolar connective tissue.
And identify the tissue based on its location.
Lamina propria.
Lamina propria.
Notice I'm basically asking the same three questions over and over again. Membrane, tissue, tissue based on location, right?
I have a question. So when we were talking about membranes, you did mention that this would be like the location would be in the oral and nasal cavities. So is the nasal cavity wrong? If we were to say that?
Technically, it's the nostril. Nasal cavity
Right.
is ciliated pseudostratified. So if you're going to use nasal cavity, that's for ciliated pseudostratified. If you want non-keratinized, really it's the external nares. It's really just the vestibule or the nostril where you would actually find that.
Oh, because it depends on the location
Yeah.
for each. So, okay.
Yeah. Plus oral cavity is much easier to say, right? Identify the membrane.
Membrane.
There you go. Sarah's got it. Cutaneous.
Yes, yeah.
Cutaneous membrane. Excellent. All right. And in fact, although we don't see it here, this connective tissue here is the areolar. Whereas if we go to the previous picture, notice this is also cutaneous membrane. This is also the skin. The areolar connective tissue is here, but notice as you look down here more, you can see the collagen fibers. So up here is the areolar, and down here is the dense irregular. All right. So notice as we look at these tissues now, we can now layer that membrane information we were talking about before onto it. All righty. We have a ton of material still to cover. So as much as I would like to end it there, we cannot. So what we need to do is we need to use the rest of our time and we are going to now, we're done with our tissues finally. And so now we get to finally talk about our our first major organ system that we are going to be talking about, and that is going to be the integumentary system. So let's take one more quick break because we have so much to cover. But again, I know that that information was dense. So actually, let's actually leave it here on this. And then we will take one more quick break. Let's come back at 11. We'll go 55. We'll take a 12-minute break. And then I will start the recording at that point. And from there, we will go on to the skin. All right. So one last chance to stretch, take a biology break, get something to eat, drink, cry, whatever it is you need to do, get it done and over with, and be back here at 11:15 to finish off the rest of today by starting our discussion of the integumentary system. I'll see you in 12 minutes. Excellent. So we finally finished with our groundwork. Time to move on to our organ systems. And the first one we're going to start with is the integumentary system, or our cutaneous membrane, or our skin. Now, as we know, all organ systems are made up of organs. And of course, what is the primary organ of the integumentary system?
The skin.
Yeah, absolutely. The skin, or what we would call the integument. Excellent. And like all organ systems, it is going to have accessory structures or accessory organs that are going to help to assist in its main functions. And what are some of the accessory structures of the integumentary system?
Epidermis.
Okay, well, that's a part of the skin. That's actually a part of the organ. I need something, other structures or other organs that will.
Hair.
Hair, what else?
Nails.
Nails and.
Glands.
Skin.
There we
Okay.
go. Glands. Absolutely. Excellent. So those are the things that are going to help. And here we see the pretty picture putting all those things together in one location. As we mentioned previously, our skin serves lots of functions. And one of the primary ones we talked about was protection. But now let's be more specific. Protection from what?
Viruses and bacteria.
Okay, so harmful pathogens.
UV rays,
Excellent. UV radiation. What else?
bacteria. Oh, here you have that.
Physical, like
Yeah.
Wear and tear.
Physical or chemical harm, right? So pouring, something caustic on your skin, falling off your bike and skidding your legs, something along those lines. Excellent. I can think of at least one really good one.
Water.
Yeah. Water loss. Whoops. Right. As we talked about, when you take that bath, you don't blow up like a sponge as a result of it. And at the same token, while we lose some water through our skin, we don't lose all of it. Think of it this way. If someone has 3-degree burns over major parts of their body, assuming they survive the injury, the two biggest fears they have is pathogens getting in and harming them, and dehydration. So notice in those cases, someone with severe burns is kept in a very humid and moist environment that is also sterile to protect them from water loss and to protect them from pathogens. Excellent. What's another function of the skin? What else does our skin do for us?
keeps us warm.
Right. Well, it plays a huge role in our temperature regulation, not just keeping us warm, but keeping us the right temperature. Absolutely, right? Whether it is sweating, whether it is changing blood flow, absolutely. What else?
Containment.
Containment of what?
Organs.
Yeah.
True, it does provide that protection, keeping our stuff on that side. But let's think about that too. Is it a completely impenetrable barrier?
No.
Semi-permeable.
Yeah, so it is going to allow some limited absorption and secretion. I know this has never happened to anybody in this class, but believe it or not, there are some individuals who will partake of a large amount of alcohol in a single sitting. Right? Often those individuals may feel bad about it the next morning. In fact, could indeed be hungover. And if they're feeling a little guilty, they may get on the treadmill to try to work out, to get rid of some of that. And as the sweat comes out of their body, what do you notice about that sweat after someone's drank a tremendous amount of alcohol?
They smell like
The
alcohol.
smell.
Yeah, you can smell the alcohol coming out of them. Like I said, I know that's never happened to anybody here in this class, but maybe instead you went
Never.
you went to that nice fancy Italian restaurant and got the 30 garlic chicken. And after eating the 30 garlic chicken, the next day you felt a little guilty, and so got on that treadmill, and you can actually smell the garlic coming out of somebody's skin as well. So there is some limited secretion. Or let's say you're trying to stop that horrible vaping habit you have. How's one of the ways you can stop smoking?
The patch.
Yeah, you put a patch on the skin that allows some lipid soluble materials to be absorbed. So yes, it absolutely is an important barrier, but it also plays a role in getting things out of the body and getting things into the body. Excellent. Two other really important functions. What are they?
Like your nervous system, like your response to obviously pain.
Yeah. Massive amount of sensory information is coming in from your skin. Yes, pain is definitely a part of it, tickle, itch, things along those lines. But also, could you reach into your pocket and just by feel pull out a quarter?
Yes.
Yeah.
Yeah, absolutely. And even though you're not thinking about it right now, if someone like me points it out to you, can you feel the weight of your shirt on your shoulders or the feel of your chair on your back and on your butt as you're sitting there?
Yes.
Yeah, you weren't paying any attention to it before, but now that I've mentioned it, you can pay attention to it and you're aware that we receive a massive amount of information from the world around us, from our skin. And one other really important function.
The skin turns the UV radiation into vitamin D.
Absolutely.
Yes, we just
Learn about that. Yeah.
did that. Yeah.
Vitamin D synthesis, absolutely. We have a special cholesterol in your skin that when triggered by UV radiation is converted into vitamin D. Vitamin D is then taken by the body and actually converted into an important hormone called calcitriol. Oops. Calcitriol. And anybody know what that vitamin D is useful for?
Doesn't it help us process calcium or something?
Yeah, not just process calcium, but absorb calcium.
Yeah.
You could drink a whole cow's worth of milk, but if your body doesn't have vitamin D, you're not going to be able to absorb most of that from your digestive system. Now, unfortunately, most of us are inside, on TikTok or doing, something along those lines. So we don't get outside a whole heck of a lot. And when we do outside, we slather ourselves with sunscreen. So we're not all getting necessarily the UV radiation that we need. So what do they do instead? They put vitamin D in pretty much everything, like milk, for instance, has vitamin D in it so that we can absorb the calcium from that meal when we're eating it. Excellent, excellent. So there you go. Those are our 5 big functions. That protection, that regulation of temperature, all that cutaneous sensation we talked about, excretion and absorption, and vitamin D synthesis. So it is very, very important function for us. Absolutely. Excellent. When we talk about the skin, and let's talk about the skin itself first, we'll talk about all of these accessory structures. As we know, this is also known as our cutaneous membrane. And that's, oops, if I spell that correctly. And that is definitely a good thing because as we know, our cutaneous membrane is made of an epithelial tissue sitting on top of some connective tissues. And so when we identify the organ that is the skin, it's our largest organ. The study of it is the field of dermatology. And it is basically consistent of just 2 parts. The superficial layer is what is known as the epidermis. And being superficial, what is the tissue type that forms it?
Mucus. Oh, the tissue type?
Yeah.
Oh my God.
Is it non-keratinized?
Is it non-keratinized?
Or no, keratinized, stratified squamous epithelium.
There you go. Our epidermis is indeed that keratinized, stratified squamous epithelial tissue. And then the deep layer, the dermis, is made up of the 2 connective tissues plural. And what were the 2 connective tissues plural that we said make up the dermis?
Areola and the irregular.
And the
Fancy, right?
dense irregular connective tissue.
Excellent. Perfect. So there you go. Now, Notice associated with the skin, but not a part of it, is what we call the hypodermis. As the name indicates, it is below the dermis. It is also referred to as the subcutaneous layer, which means below the cutaneous membrane. And if you're below the cutaneous membrane, can you be a part of the cutaneous membrane? Not a trick question.
Yes.
If you're below the boat,
Oh,
can
no.
you be a part of the boat?
No.
No. So this is not a part of the skin. It is made up primarily of areolar and adipose connective tissues, but it's not a part of the skin. But that doesn't mean it doesn't play an important purpose. What is the function of our hypodermis? Well, what's my subcutaneous fat doing for me?
Protecting.
Yeah, that's what I keep telling my wife.
Friction.
I'm not fat, I'm just protecting my internal organs. Absolutely. Insulation, helping in temperature regulation. Absolutely. However, notice I also need it here in my forearms. Now, there isn't a lot of fat here in my forearms, but there is areolar connective tissue. So that areolar connective tissue here, is it necessarily providing protection or insulation here on my forearm? No. So what is it doing? Well, Halloween is right around the corner. And I'm sure like you, we all have our favorite things, our go-to costumes that we like to dress up with. And if I don't have anything particular clever to do this year, then I'll go to my standby, which is where I put on a pair of short shorts and I paint my entire body green and go running around like the Hulk. And it looks awesome for about the first 30 minutes. But as the paint dries and as I continue to move my arms and legs, what happens to that paint?
It crackles.
It cracks and it breaks.
Yeah.
Do we want our skin doing that?
No.
No. So notice our muscles are changed shape. In fact, that's where they get their name. Muscle comes from the Greek word mus, which means mouse, because when you contract your muscles, it looks like little mice running around underneath the skin, right? And as they change their shape, they put pressure, they put stress on the skin. Notice that paint doesn't have any give. So when I put the when I stretch, that paint cracks, that paint breaks. We don't want our skin to do that. And that's what the hypodermis does. Notice the hypodermis loosely connects the skin to the muscle underneath. So I can actually move the skin around on this above my muscles because of that subcutaneous layer. So again, it's not a part of the skin. That doesn't mean it's not important. It is absolutely important. It plays an important role, like I said, in that protection, in insulation, and probably most importantly, it loosely connects the skin to the tissue underneath to help to maintain the integrity of our skin. All right. Let's go back to our picture for a second. So notice again here, as we look at this illustration, we have this layer here, which is our epidermis. This layer here, which is our dermis. And then underneath that, we see the adipose making up our hypodermis, that layer underneath. What we're going to do now is focus solely on this tiny little superficial layer that is our epidermis. So that's where we are going to focus. Epidermis, of course, as we know, is a keratinized, stratified, squamous epithelial tissue or epithelium, which of course means that it is made up of densely packed cells. So if we're going to talk about an epithelial tissue, we have to talk about the cells of that epithelial tissue. And there are 4 primary cells that are found in our epidermis. The first, and by far most common, making up well over 99% of the cells of our epidermis, are cells called keratinocytes. And what do you think a keratinocyte does?
This is keratin.
Yeah,
Makes
exactly. Keratin is or really what it makes is these keratohyalin granules, proteins that are put together into bundles to make keratin fibers. These keratin fibers are what make our skin tough, are what make our skin hard. This is what provides that strong protective outer layer. But remember, we also need the skin to be
Isn't there like
water.
an overproduction?
I'm sorry? I didn't
Can't
hear that.
an overproduction of keratin cause like?
Uh,
Can't an overproduction of keratin cause keratosis pilaris?
Yes, so there are a couple,
In
that
the
can
skin?
occur. Dandruff, for instance, psoriasis, all of those are conditions where you're either overproducing or producing abnormal keratin, absolutely. So those things can be associated with this, absolutely. But in normal healthy cells, they produce that tough fibrous protection. However, remember, we also need the skin to be waterproof. And so what these keratinocytes also produce are these lamellated granules. These basically is like a car wax that fuses the cells together and makes them waterproof. Makes it so that no water can get into the cells, but also no water can get out of the cells either. Now, if we fill these cells with keratin, so much so that they get rid of their nucleus and everything else, and then we fuse them together and waterproof them so cells and water and oxygen and nutrients can't get in, waste can't get out, what's going to happen to those cells?
Will they die?
Yeah, and of course, what do you know about the superficial layers of your skin?
They're very
It
keratin.
dies.
Yeah, they're basically dead keratinocytes, absolutely. Fused together dead keratinocytes. So much so that, as we know, when we burn, they don't just slough off, they peel off as a sheet, right? Because they're so stuck together.
What are granules?
Granules are basically just dense vesicles that contain a lot of substance, and typically they're dark enough we can see them under a microscope. So
Okay, so they look like
they're
specks.
yeah, they're vesicles that contain specks inside of them, absolutely. So make up more than 99% of the skin. So if you were to throw a dart at the epidermis, well, A, it would hurt, but B, it most likely would hit a keratinocyte. However, there are some other functions of the epidermis. And so for that, there are 3 specialized types of cells that help our epidermis do what it needs to do. Probably the one you're most familiar with are the melanocytes. What do melanocytes do?
Produce melanin?
Yeah, that's the stuff that makes you sleepy, right?
No.
What's the stuff that makes you sleepy then?
Melatonin.
Oh, melatonin.
Melatonin,
Okay. I always get those two confused. Yeah. So what is melanin? What does melanin do then?
It creates the pigment in your
Yeah.
skin.
That is the pigment of our skin. One of the interesting things, and again, only a couple people are on camera, but we've seen most of the people in the class at one point or another. Every single person in this class has essentially the same amount of melanocytes in their skin. But is every single person in this class the same coloration to their skin?
No.
No.
Isn't that you have more melanosomes?
Right, exactly. So there are 2 ways that are different. One is the actual color of the pigment that is made, and the other is the amount of it. Absolutely. So there's variations in all of those things, which vary.
Eumelanin and eumelanin and phomelanin, something like that.
In fact, there are over 4 genes involved in making the color of our melanin, and they are each of them has at least 2 versions, a light and a dark. Some have more than 2 versions. So again, we are truly a spectrum of colors because of the differences in the amount and the color of the melanin that is produced. One of the interesting things about these melanocytes, there's actually two really interesting things about the melanocytes. The first is that they are located very deep in the epidermis, down near the bottom. And their job is basically to make that melanin, bundle it up into a vesicle, and give it to the keratinocytes that are next to it. Then what those keratinocytes do is basically take those melanin granules that you were talking about and use them to basically form a shield, an umbrella that protects them from the UV radiation. So they make this pigment, give it to the keratinocytes to protect the keratinocytes.
Is that called like a superficial cap or something like that?
It's basically just a protein shield that it's putting over the surface of it,
Oh.
providing that pigment, providing that protection. Very, very useful for protecting us from UV radiation, because as we know, UV radiation can damage our DNA. And if we damage our DNA in just the right, or really just the wrong way, right, then suddenly that cell could start producing abnormal proteins, divided uncontrollably. And what would we call that condition?
Skin cancer.
Yeah, it'd be skin cancer, absolutely. But here's the interesting thing. The melanocyte makes all this melanin and gives it to the keratinocytes, but it doesn't keep any of it for itself. Now, at least it's deep, so it's more protected from the UV radiation, and the keratinocytes that are protecting themselves can help to protect the melanocytes. But the melanocyte doesn't actually have its own protection. So if that UV radiation gets down to the nucleus of our melanocyte and causes it to mutate abnormally, right? What kind of skin cancer do we call that?
That carcinoma?
Well, not a carcinoma, it's a melanoma.
Oh, melanoma.
And not that anybody wants any kind of skin cancer, but if you had to have a skin cancer, would melanoma be the one you would want?
No.
No.
Yeah. Melanoma is an incredibly aggressive. In fact, it can often and sometimes kill within months of diagnosis. It is a very, very dangerous, very aggressive form of skin cancer. So ironically, it's protecting everybody else, but doesn't protect itself. Right? Which is a
Are we
good
able
thing.
to see this under the microscope?
Yep.
Okay.
And we will do that later. Absolutely, we will see that. So we will be able to easily recognize both of these under the microscope. And actually, let's do that now because we're running out of time. Let's do this now before we forget. Perfect. So here, remember before we saw our thin skin, and let's just say for argument's sake, on the exam, I were to point an arrow, there's a little small, an arrow here and ask you to identify that cell. What do you think your answer is?
Keratinocyte.
Keratinocyte, and that one,
Keratinocyte.
and that one. Keep going.
Keratinocyte.
And that one.
Keratinocyte.
Keratinized.
Keratinocyte, and that one, and this one, and this one, and this one. What are all of these cells?
Keratinocytes.
Excellent. All of these are keratinocytes. All right. However, remember one of the things that we talked about, the melanocytes, there are two things we pointed out. One is that they're very deep in the epidermis. And two, while they make pigment for the surrounding cells, they don't keep any of that pigment for themselves. So notice down here, there are these cells that have very clear cytoplasm, very deep in our epidermis. All of these cells that I have circled are great examples of melanocytes. So the way you're going to recognize a melanocyte is it is deep in the epidermis and has a very clear cytoplasm. makes it obviously and noticeably different from all the keratinocytes that are everywhere else. Okay. Now, there are two more types of specialized cells in the epidermis, but I'm not going to make you recognize them histologically, right? If I asked you an essay question, describe the 4 types of cells, you would need to identify them and talk about them, but you're not going to need to recognize them on the microscope. The first of these are what are known as epidermal dendritic cells, also known as Langerhans cells. And of course, why are they known as Langerhans cells?
Because you founded it.
Yeah, good old Bob Langerhans was the first person who described it and planted his flag in it. Again, we're slowly starting to move away from this, and these are also called dendritic cells because a dendrite basically means a small process. And notice this cell has these small processes that come off of them. These dendritic cells are basically similar to white blood cells that we would find in our blood, where they provide protection. They're also similar to one other cell we've talked about. They're also similar to mast cells. And what do mast cells have in them? We just
Is
finished.
it
talking-
mucus?
Yeah.
Oh, his.
Remember, mast cells contain histamine. That histamine, remember, plays an inflammatory response. If you get poison oak or poison ivy on the surface of your skin, that irritation causes redness, causes hives to occur because of these Langerhans cells releasing that histamine, causing that immune response. So these Langerhans cells contain histamine. They're also phagocytes. What did phagocytes do again? We talked about these when we talked about lysosomes. Remember phagocytes?
They store,
Well, they don't store so much.
They break down, right?
Yeah,
Like,
exactly. They find
they
some
get-
harmful pathogen and they bring it inside and then they gobble it up, break it down so that that virus or that bacteria doesn't hurt you anymore.
Mhm.
So these play a very important role in our immune response. What's interesting about these is they can actually move through the layers of the epidermis. So while they're commonly in the deepest layer, they can move around. So we'll just say deeper layers, plural, because they can move around within the deeper layers of that. The last cell we need to know is a tactile cell. That tactile cell is also sometimes referred to as the Merkel disc. And of course, why is it called the Merkel disc?
Because Merkel discovered it.
Good old Bob Merkel was the first one who discovered it and planted his flag in it. These tactile cells, they provide us with our fine discrimination. Remember before I mentioned you could reach into a pocket full of change and pull out a quarter. How are you able to do that?
Sensory information. or-
Yeah, but specifically, what specific sensory information allowed you to tell a quarter from everything else in your pocket?
Size.
Size is one of the things. And what else is special about a
Texture.
quarter? Yeah, that ridge on the outer surface. It has that ridge on the outer surface that you can feel. That ability to feel those fine ridges are something that you could easily disguise. Think of it this way. If I had a nickel and a quarter and I rubbed the edge of it on your finger, do you think you'd be able to tell the difference between the edge of a nickel and the edge of a quarter?
Yes.
Yeah.
Yeah. But what if I did the same thing on your back? On your back, do you think you'd be able to tell the difference between a quarter and a nickel?
No.
Probably
Okay,
not.
Maybe slightly, but not really.
Maybe, it would depend, I guess, maybe on how hard I pushed, maybe, or something like that, but it'd be a lot harder. So notice these tactile cells are all over our body, but are they equally distributed throughout the body?
No.
No. Where do you think we have the heaviest concentration?
Our hand.
Digits, like our toes or our fingers.
Good guess. Definitely the hands, especially the thumbs. But there's one other area as well. It's actually not the feet.
Is it
Our face.
mouth?
Face, especially the mouth.
Yeah. I'm-
Like
Absolutely.
the tongue.
And again, for those of you who are about to have children, you're about to learn this. How does a baby explore the world?
Their hands.
Yeah, it grabs things, and as soon as it grabs it, what does it do with it?
Straight to the mouth.
Shoves it in its mouth. Absolutely. Because that's where it has most of these tactile sensors. So that's how it's exploring its world, grabbing things and shoving them in their mouth, because that's where we have the most number of these tactile cells, providing us that fine discrimination, that fine ability to be able to feel. Yeah, we have them on our forearm and our small of our back and back of our legs and other places as well, but not nearly as many. All right. Excellent. All right. Last thing I want to do for today, and then we will from there be able to finish this on Wednesday. When we look at that epidermis, that tiny little piece of the skin, there are actually either 4 or 5 distinct strata or sublayers to that layer. And whether it's 4 or 5 is whether it's thick skin or thin skin. Thick skin has 5 layers, and thin skin has only 4 layers. Where do we find our thick skin?
Bottom of our feet.
Bottom of our feet, and
Elbows?
no, not a bad guess. That is a good guess, but no.
Our hand. No.
Yeah,
Our hands.
palms of our hands,
Okay.
soles of our feet. Right? Think about that. The front of your hand and the back of your hand are very different from each other. Yes, one difference is the thick skin has 5 cell layers, whereas a thin skin has 4 layers to the epidermis. But what's the other big difference between thick skin and thin skin?
Your
look at the-
sensations?
Look at the front of your hand, look at the back of your hand. What do you notice that's different about them?
You don't have the fingerprint ridges.
Ridges, excellent. So thick skin has ridges, whereas thin skin does not have ridges.
I mean, our knuckles are kind of rigidy. That's a word. I don't think it is.
Well, I think, but again, it's only because the skin is clustered together, right? It's bunched together. If you stretch it out, you don't see them.
I know we can't see this, but the keratinocytes, there's probably going to be
You're
less
right.
on our back.
There's more here. You're right. Thicker skin has more keratinocytes. You're right. We can't really see that, but you're missing the obvious thing you can see. Again,
Pigmentation.
difference can be pigmentation. What else? There we go. Sarah's got it. Sorry, Sarah, I didn't catch it. No hair. Thick skin has no hair on it, right? No matter what type of activities you participate in home, no hair is going to grow in this region of your body, right? Thick skin has no hair. It is hairless skin. Whereas thin skin has hair. So absolutely, those are some of the big differences between the two of those. We've actually looked at this already. Here we've seen thick skin, and here we see that thick skin like we looked at it before. And notice the thick skin has 5 sublayers to it. Now, one of the interesting things about the skin is when we identify the sublayers, instead of going from superficial to deep, we actually go from deep to superficial. And the reason we do that is because that is how the cells grow. They start down at the base where they're first formed. And then from there, after they're formed, they're pushed up. They fill up with keratin, they die. And then when they get to the top, they slough off and become that dust in your apartment. So when identifying this layer, there's always important to have useful mnemonics to help to remember the 5 sublayers: stratum germinativum, stratum spinosum, stratum granulosum, stratum lucidum, and stratum corneum. And I will tell you right now, the stratum germinativum is also sometimes referred to as the stratum basale. You are welcome to use either term when you are identifying this layer. Basal is easier to spell, so if you want to use that, I'm totally fine with that. And it also helps with the mnemonic. Mnemonics are, of course, tools that can help you to remember material. I've heard many good mnemonics. However, my favorite one, and the favorite ones are often something that's stupid, but mnemonics usually use the first letter of a word to help us to be able to remember that. And my favorite one, I had a student come up with this one, I got probably 7-8 years ago, but it's the one that stuck with me. Again, from bottom to top: boys stink, girls like candy. I don't know why that's the one that stuck with me, but that is the one that has. So stratum basale, stratum spinosum, stratum granulosum, stratum lucidum, and stratum corneum. So also, as I mentioned, thin skin is the one that lacks the stratum lucidum. So the stratum lucidum is thick only. All right, so with that piece of information, what we are going to do on Wednesday is we are going to start our discussion of the 5 sublayers, what's going on, how they grow, how they form, and we're going to move our way up from there. All right, any questions on any of that?
So you said for the epidermis, the cells, the 3 and the 4th, the epidermal and the tactile, they're not on the lab, but they'll be on the lecture possibly.
So if we go back to a picture like this, notice on a picture like this, we can clearly identify a melanocyte. We can clearly identify a keratinocyte. Is it possible histologically to see a dendritic cell or a Merkel disc, a tactile cell?
No.
Yes,
No.
it is actually
Oh.
possible. However, it's very hard. And is it something that's worthwhile to force you guys to try to figure out to do for possibly one question on the lab exam?
No.
There's far better use of your time. These 2 are really obvious, so histologically you'd have to be able to do that. But I'm never on a lab exam going to point at a piece of tissue and say, what is this cell? And you're going to say a tactile cell. However, could I ask you on an essay question on the lecture exam to identify all the cell types found in epithelial tissue? And when you're discussing that, would you need to talk about tactile cells and dendritic cells? Yeah, absolutely.
Yes.
So I would ask it in something like that, but I'm not going to on a histology slide. So you're not responsible for them on a histology slide. Okay, you need to know what they are, you need to know what they do, but I'm not going to point to them on a piece of tissue and ask you to recognize them. It's too hard. It's possible, it's just too hard. All right. Great questions. Any others?
I have a
I
question.
Yep,
Go
go ahead.
It's a, so is the integumentary system that we just learned, that's all going to be on the next test, correct? It's not going to be on this current test.
No, it's going to be on the test on Monday. Yes. Everything we talk about today, everything we talk about on Wednesday, the entire integumentary system will be on your exam on Monday. Yeah. And so again, if you think about the way this class has been divided, this next test is basically going to have 3 parts: epithelial tissues, connective tissues, and integumentary system. So while it's not perfect, it's going to be a pretty even distribution of points between those 3 things, because those are the 3 main topics that we've had for this section, and we've been able to spend about an equal amount of time on each of them. We're spending about a lecture, and a change on each of these 3 topics. And so that's going to be the way it's going to be broken down.
Got it.
Okay. All right, excellent. Was there one other question? I thought someone else was asking a question as well.
I was, but I think I kind of answered it. I was going to ask if the 4 cell types were solely in the epidermis and nowhere else.
Correct. So far, we're only talking about the epidermis. When we get to
Okay.
the dermis, there will be other cell types there.
Okay, thank you.
Excellent. All right, and we're building to that. But so yeah, we've got a ton of work to do on Wednesday. You have the lecture slides. There's no reason that you shouldn't look at this material ahead of time to be able to do that and prepare that. And let's go ahead and stop the recording.